Good morning, everyone. Good morning and welcome. I'm Andrea Bayer, the Deputy Director for Collections and Administration, and I'm delighted to see so many of you here today for what promises to be a fantastic symposium. The Met plays an essential role in advancing the study and appreciation of cultural heritage, a mission that extends far beyond the museum's walls. We're privileged to have a dedicated team of experts spanning conservation, curatorial, and scientific research, enabling us to conduct in-depth studies of the materials that compose the masterpieces from all parts of the world in our collection. With these resources, the Met is uniquely positioned to tackle some of the most pressing and complex questions in the field. The work presented today is a testament to the power of this interdisciplinary collaboration, exemplified by the ambitious project focused on chia oil, an endeavor marked by its extensive partnerships with both international and local institutions. This work has important repercussions in our understanding of artistic practice in Mexico and its connections to European and Asian cultural history. Our research has been enriched by the contributions of a diverse team of artisans, art historians, archaeologists, conservators, and scientists, highlighting the importance of cross-disciplinary approaches. The project underscores the significance of identifying and understanding the materials in our collection, even when the challenge is formidable. Organic analysis, in particular, presents unique difficulties due to the variability of the natural materials involved. With numerous plant and animal sources and complex, often undocumented preparation methods, these substances further complicate research as they deteriorate with age. As an exploration of organic binding media, today's presentation deepens our understanding of drying oils, thereby challenging the traditional notion that oil painting is solely a European innovation. To address tough questions, one must have the right strategy. In this case, a groundbreaking approach for investigating biological materials elevates our traditional oil analysis techniques by integrating cutting-edge lipid, protein, and genetic methodologies. This innovative methodology not only enhances our ability to analyze known substances, but also paves the way to exploring previously uncharted biological material and artworks. Although this project began with a focus on Mexican colonial period, its significance goes beyond temporal and geographical boundaries. It exemplifies how thinking outside the box allows us to apply these methodologies on a global scale, making this approach adaptable for various inquiries where biomolecules are critical for determining the identification, provenance, or authenticity of artworks. We'd like to formally acknowledge the diverse sources of support that made this research possible. The National Endowment for Humanities generously funded our research activities and this public symposium. Additionally, funding from the MEXAM Foundation facilitated the participation of our international speakers, while a generous donation for scientific research partnerships through the Mellon Foundation enabled the simultaneous translation for this event. This pioneering project was anchored by the exceptional team assembled by my colleagues Julie Arslanoglu, Jose Lazarte, and Rhonda Castle. We also wish to recognize the significant contributions of ARCH, our CNRS-endorsed international laboratory in partnership with the University of Bordeaux, which played a crucial role in advancing this research. Furthermore, we extend our gratitude to our collaborators at the Hispanic Society Museum and Library, the American Museum of Natural History, Weill Cornell Medicine, and the City College of New York. Can't tell you how happy it makes me to read all these names aloud of the people that we've been working with on this project. We are especially thankful to the numerous Mexican museums and institutions that gave access to their important collections, and to the lacquerware artists who generously shared their knowledge and expertise with us. We are looking forward to hearing some of the results of all of this research. Thank you very much for being with us today. Enjoy the symposium, and I pass on to Jose. Thank you very much.
Okay, good morning, everyone. My name is Leisha Migichi, and I'm an associate research scientist here at the Met in the Scientific Research Department. Today, I have the immense pleasure of moderating this morning's session. And before I get into our introductions for our first speaker, I want to make an announcement that today's session will be simultaneously delivered in Spanish and English. If you are in need of the Spanish language translation, please make sure to grab a headset by the back of the room. Now, I realize I said it in English, so hopefully someone can translate. Louder? Okay. So the first session today is from historical evidence to novel scientific capabilities. Our first speaker is Jose Luis Lazarte, who is an associate conservator in paintings conservation here at the Met. Jose Lazarte holds a master's in science from the Winterthur University of Delaware graduate program in art conservation. During his academic career, he gained experience through internships at the Yale University Art Gallery, the Prado Museum, and the Fine Art Museums of San Francisco. Jose joined the Department of Paintings Conservation at the Met in 2016 as a Mellon Fellow and transitioned to a staff position in 2019. His work focuses on the treatment and technical study of European and American paintings from the 16th to 18th centuries, with a particular emphasis on artworks from Latin America. In addition to his conservation practice, Jose has published technical research on new Spanish paintings and conducted conservation workshops in Peru and Argentina. So with that, I'd like to open the floor to Jose.
There you go. Thank you, Alicia, for that kind introduction, and thank you all for joining us today. Before I jump into my talk, I would like to echo previous acknowledgments and extend our warmest thanks to the National Endowment for the Humanities for their generous support through a grant that we received in 2022. The NEH support allowed us to create partnerships with different local and Mexican museums that graciously opened their doors and their collections to us. The Hispanic Society Museum and Library has been a key partner in our project since the very beginning, and if you haven't done so already, I urge you to visit their museum in Hamilton Heights. We're also extremely grateful to the Met Scientific Research Partnerships for supporting the Expert Study Day that we are having tomorrow at the Hispanic Society, and to the Mexican American Cultural Foundation, which made the participation of international speakers a reality today. Since 2013, the Met has made concerted efforts to collect, study, and display Spanish American artworks within its galleries. Over the last decade, we've had the unique opportunity to deepen our understanding of these works through careful restorations and technical research. This morning, I'm excited to guide you through our journey, examining the use of chia oil as a paint binder in the Viceroyalty of New Spain, present-day Mexico. I will begin with the historical evidence of its use in oil paintings, the experiments we conducted to qualify the physical and mechanical properties, and end with our focus on the output of two artists that are said to have used chia oil, the lacquerware master José Manuel de la Cerda and the easel painter Juan Correa. In 1715, the renowned Afro-Mexican artist Juan Correa was commissioned to search for art supplies on behalf of a merchant in the Caribbean city of Campeche. With his extensive experience as a painter, Correa would have been well acquainted with the best sources for such materials. Apothecaries and specialized vendors scatter across the lively streets of Mexico City. The document displayed here is the merchant's inventory, listing the materials Correa was instructed to procure. These include brushes and common pigments such as verdigris, cochineal, minium, vermilion, and indigo, among others. Additionally, the list specifies diluents like fur-based turpentine, referring the list as aceite de abeto, and of note, two drying oils used for paint preparation, the widely known linseed oil, and the lesser known oil native to Mexico, chia oil. The request for chia oil from Mexico City's most prominent painter, Juan Correa, holds significance considering the massive 29 by 25 foot canvases he painted for Mexico's cathedral. It is clear Correa had expert knowledge of oils and the properties, and that he would only be asked to procure materials he was deeply familiar with. This raised an intriguing question among scholars, including colleagues such as Pablo Amador, Elsa Arroyo, and Desiree Moreno. And the question was, did Correa use chia oil in his own works? This inquiry led us here at the Met to explore the identification of local oils in New Spanish paintings, as well as the implications of their use and their conservation requirements. Chia oil, along with the fat from the parasitic female insect known as aje, has been used in the production of Mexican lacquerware since pre-Hispanic times to create water-resistant surfaces on gourds, and the technique endures to our days. During Correa's time, chia oil was used to create reflective finishes on wooden furniture and decorative objects, as shown in the middle of the screen, in the middle of that timeline. I'll discuss these black lacquer objects, which I'm trying to point to, but it doesn't work too well, later on in my talk. During the Viceregal period, Mexican lacquerware evolved based on its city of origin. In Uruapan, Periban, and Pátzcuaro, three cities in the state of Michoacán, both chia oil and aje were mixed. In contrast, Olinala, located in the state of Guerrero, relies solely on chia oil. In both regions, these oils were combined with minerals and pigments and polished on surfaces to create rich lacquer layers. The lacquer practices in the cities not only varied in the binders used, but also in the methods employed to achieve their vibrant polychromies. In Periban, artists incised motifs into the lower lacquer layers, filling figural voids with contrasting colors of lacquer. Pátzcuaro featured a bastiglia-like technique made with a resinous bowl applied over an evenly polished lacquer ground, which was then painted and gilded. This technique is most akin to Japanese European imitation of Asian lacquerware. In Olinala, layers of polished lacquer were overimposed and meticulously cut away one by one to reveal the contrasting colors beneath. These variations that you see on the screen show the intricate nature of the lacquerware tradition, and what I provided is an overall view of the processes involved. During the 17th and 18th centuries, local oils from chia and Mexican poppy seeds, known as chicalote, are said to be commonly used alongside European linseed oil. Chia oil has a rich history in Mexican lacquerware, but it's also noted in relationship to paintings. In his Chronicle of New Spain from 1575, Francisco Cervantes de Salazar wrote that, quote, "Chia oil is of great virtue. If a painting is covered with it, it will preserve the vibrancy of its colors and protect them against humidity," end quote. Cervantes likely refers to chia oil as an oil resin varnish, which was traditionally used to coat paintings for saturation and protection. In 1784, Casimiro Gómez de Ortega, director of the Royal Gardens in Madrid, mentioned chia while expanding on the work of the 17th century botanist Joseph Kerr. He stated that, quote, "Salvia hispanica is cultivated in La Mancha and other provinces of Spain, and in Mexico, where it is called chia, it is a perennial plant. From chia seeds, an excellent oil is extracted for varnishes and paintings, and it is used for this purpose in all of New Spain, where it is preferred to others," end quote. Around the same time, there were attempts to produce chia oil in Europe. Francisco Javier Clavijo, a Mexican Jesuit priest exiled in Bologna, cultivated chia plants with the intention of making its oil known to Italian painters. But he lost his plants to the first Italian frost. In 1788, the new Spanish proto-scientist and historian José Antonio de Alzate y Ramírez devised an experiment to distinguish between paintings from Spain and those from Mexico, claiming that the identification of the oils used would determine their place of origin. If a painting contained linseed or walnut oil, it would be European, and if chia oil were present, it would be Mexican. For his test, Alzate would burn a small piece of paint removed from the edge of the canvas, identifying the oil by using his sense of smell. In Alzate's opinion, chia oil was the oil of choice in Mexico and equivalent, if not superior, to linseed and walnut oil. Overall, the archival records I presented here and before show that chia oil remained an alternative oil in artistic circles and that it was championed as an exportable Mexican crop. The need for a substitute for linseed oil in Mexico may be explained because flax, the plant used to produce linen and linseed oil, was not widely grown in Spanish America. During the reign of Philip II, in the early 17th century, the Spanish monarchy prohibited its cultivation to protect European trade. This ban wasn't lifted until the late 18th century, leading scholars to believe that the linseed oil used in Mexico was invariably imported from Europe. Going back to the list, what is surprising is the amount of chia oil Correa acquired in Mexico City. 2 botijuelas, about 12 liters of oil. What is also telling is that Correa could not procure linseed oil as requested and supplemented the order with chia oil. This suggests that Correa thought the oils were interchangeable or at least compatible. To understand this concept, we studied the physical and mechanical properties of chia and linseed oil to determine how their handling properties compare to each other. We traveled to Michoacán and Guerrero to interview the artists that are currently revitalizing lacquerware traditions in their home studios to see how chia oil is extracted. Later today, you'll witness how this tradition endures through a live demonstration of lacquerware master Martina Navarro and her family. And you see Martina in the middle of the screen, and she's sitting right there. In Olinala, we were welcomed by Paula Escudero Mejia, who no longer makes lacquerware but makes large batches of chia oil for artisans working in the city. The process of expressing the oil from the seeds has 5 basic steps. First, chia seeds are toasted. Then the seeds are ground into a meal and kneaded with incremental amounts of water until it feels oily to touch. Lastly, the chia dough is shaped into balls, wrapped in muslin, and pressed in a metal frame to extract the oil. Through our visits to other workshops, we also learned that some artisans use modern oil extractors to expedite this labor-intensive process. Upon our return, we team up with 2 exceptional summer undergraduate interns, Ella Madura and Enya Velusovic. Ella worked at the Met preparing various chia and linseed oil recipes, while Enya focused on analyzing the rheology of these oils under the supervision of Professor Raymond Tu in the Chemical Engineering Lab at the City College of New York. During our research in Mexico, we discovered that each artist modified the raw chia oil using unique methods tailored to their artistic practices. Drawing from these insights, we, as well as historic treatises by Antonio Palomino, Theodore de Mayerne, and Giambattista Volpato, we developed guidelines to replicate 18th century oil treatments. To standardize our raw oils, we employed a modern extractor for chia oil while using commercially unmodified linseed oil from Kramer Pigmente. Both untreated oils were then heated incrementally up to 135 degrees Celsius. Using a whole clove of garlic as a time indicator, as described in the Spanish treatises. Before the garlic clove reached a uniform golden color, we added tiny amounts of menium, a lead-based red pigment traditionally used to accelerate drying. We also incorporated ocote, which is a resinous pine wood, and barite concentrations of 2.5% and 5% of fat from the aje insect, which we procure from the artisans in Mexico to investigate specifically its role in lacquerware traditions. You're seeing images capture every 5 minutes of linseed and chia oil heated with garlic and 2.5% aje fat. This shows that chia oil foams more than linseed oil, possibly due to the higher water-soluble content within the oil. Following 17th and 19th century purification techniques, we wash both oils in separation funnels with double the amount of water for a period of 8 weeks. Over time, the linseed oil layer separated more easily, while chia oil formed what we call a stable emulsion, likely due to the interaction of water molecules with the polysaccharides and proteins in the mucilage expelled by the seeds themselves. Enya's experiments at CUNY measured the bulk and interfacial rheology of these oils. Rheology studies how a fluid's viscosity changes under varying shear pressures, providing insights into their handling properties. At first, we saw that chia oil had a higher viscosity than linseed oil, but over time, with manipulation under high pressure, it reached a similar consistency, making both oils suitable for applications in lacquerware polishing or pigment preparation. In the following presentations in this session, you will discover the innovative analytical approach that my colleagues developed to discern between these two oils within historical paint samples using advanced lipidomic, proteomic, and genomic analysis. Now I am going to focus these last minutes on the creative process, on the techniques employed by Jose Manuel de la Cerda and Juan Correa without delving too much into the nature of the oils they used. This lacquerware tray, known as Abatea, was acquired by the Met in 2020 and can be seen today in Gallery 600. With a striking diameter of 42 inches, it was carved from a single piece of American cedar. The elaborate designs that you see on the front, influenced by Asian and European motifs, were masterfully executed by the indigenous nobleman and lacquerware artist from Pátzcuaro, José Manuel de la Cerda. This distinguished piece was likely commissioned by the Viceregal Court in Mexico City in the mid-1760s. To understand the structure of its lacquer layers and de la Cerda's techniques, we conducted cross-section microscopy. On the right, you see a magnified sample from the tray under ultraviolet light and elemental distribution mapped in false color through scanning electron microscopy. And this was done by Federico Caro here in the museum. The black lacquer ground was built in with two campaigns of a ground layer of kaolini pigmented with vermilion and lead white, followed by a silicon-rich aluminum clay and a final thin application of bone black. This sequential stratigraphy which is basically consistent across the front of the batea, demonstrates de la Cerda's pursuit for an exceptional, smooth, reflective finish. After building up the lacquer layers, he used a robust bowl. Oop, did we lose? Can you hear me? Yeah. How do you?
They're working on it. Maybe to describe it.
Yeah. Okay. After building up the black layers, no. I'll try to project.
Can we use one of these?
Maybe. Can I use one of these?
Yes.
So. Okay. Hello, can you? No. Okay, I'll just go through and after building up the black layers, he used a robust, bold mixture to create intricate filamentous designs before painting the motifs that define his style. You can see those filamentous lines here on the screen. And I don't know if my pointer works, but right there. And you're seeing this image in raking light. Our pigment analysis using XRF, Raman, FTIR, oh, there you go, FTIR and SEMEDS reveal a typical 18th century color palette. While a similar exquisite desk signed by de la Cerda was on loan at the Met from the Hispanic Society, we had the unique opportunity to study it alongside the batea. A sample from a yellow-brown area on the desk, on the desk's left Bombay drawer, show a different lacquer structure. Unlike the batea, the desk features a heterogeneous ground layer over the surface. In the upper section of the desk, which is this section that you see here sitting on top of the table, we detected a white ground composed of gesso and lead white, followed by a pigmented oil-bound thick kaolini layer. A reason for the different ground structures that we saw throughout the desk might be explained by the variable quality of the woods used in its creation. This last spring, we returned to Mexico to establish research partnerships and study more works attributed to de la Cerda and his workshop, as well as paintings by Juan Correa, since the Met does not currently hold any of his works. Thanks to the support of these institutions, we analyzed 7 additional pieces related to de la Cerda, including a remarkable panel from a now lost desk at the Museo del Virreinato, which is decorated with similar pictorial elements as the Met batea and the Hispanic Society's desk. And a vibrant red jewelry box from the Franksmeyer Museum, which you see on the right. Our findings, soon to be published, will provide the first technical analysis of de la Cerda's output. We also examined 11 paintings by Juan Correa, including 2 large biombos or screens from his later career. While we do not have scientific data on how Correa used chia oil, whether as an extender for linseed oil to mix with specific pigments or in his red preparatory layers, his influence on subsequent generations of 18th century painters is undeniable. If he were using chia oil, it would have been noted and potentially emulated by his contemporaries. Thus, the study of chia oil should not be confined to Correa and his workshop alone, but extended to broader artistic practices of the time. The historical references about the use of chia oil in paintings throughout the century show a specific need to popularize the material, and that these efforts would have been futile if chia oil did not perform as well as linseed oil to produce paintings. The work that I presented today was made possible by the generosity and dedication of many colleagues featured on the screen, and I extend my deepest gratitude to them. Thank you for your attention.
Okay, so we're going to hold all questions until the question and answer session at the end of this morning's session. So we'll move right on to our next speaker, Julie Arslanoglu. Okay, long O, no G. Okay, so Julie is a research scientist in the Department of Scientific Research at the Metropolitan Museum of Art since she's joined in 2006. She investigates the identification, interaction, and degradation of natural and synthetic organic materials, including paints, coatings, and adhesives found in artworks from all cultures and eras, using mass spectrometric, imaging, immunological, and genomic techniques. Her education and career bridge the conservation and science worlds as she holds graduate degrees in both organic chemistry from the Pennsylvania State University and in paintings conservation from the Courtauld Institute of Art. Julie is a recipient of grant awards from the National Science Foundation and the National Endowment for the Humanities, who has funded today's session, to study the impact of age, pigments, and environmental conditions on protein-based paints and their identification. And in 2022, NEH funded Julie's work to develop a novel three-pronged approach towards the biomolecule analysis for the identification of unknown artistic materials applied to the use of chia oil in art from New Spain. Julie has previously held positions at the Getty, the V&A, the University of Texas at San Antonio, and the National Institute of Health. She is co-founder of Art Biomatters, a coalition of scientists, conservators, historians, and curators focused on biological materials used to create cultural heritage, and ARCH, the Art and Cultural Heritage Natural Organic Polymers by Mass Spectrometry, a partnership between the Met and the University of Bordeaux, studying the material dimension of museum collections on a molecular level using mass spectrometry. And on that note, I'll turn the floor over to Julie.
Thank you, Alicia. So today's morning session is a little bit like a book. Each one of us will be presenting a chapter, so it'll be a little bit different from a traditional talk. So Jose's already given you a very good background about how he became interested in chia oil. And as he mentioned, the Met began collecting in around 2013. And so here I'm showing you our first acquisition of a Mexican painting on canvas by Jose Sanchez, and then our first Mexican lacquerware object by Jose Manuel de la Cerda. The Jose Sanchez was a contemporary of Juan Correa. So if there's speculation that Juan Correa was interested in using chia oil or was using chia oil, Jose Sanchez would have been part of that circle. And so we have this painting with this question, and then we have a lacquerware object from Michoacan, and we know that it was made with chia oil. So this is our starting point. I'd like to say a little bit about oils in the collection. So you can find oils in all parts of our collection. For example, you can find oils as food residues in many of the containers, the pots, the amphora that we have in the museum. You'll also find it used for paintings, not just in European paintings, but in paintings throughout the world. You can find it in furniture as a coating, but also as decoration. And then you can find it in objects that have more cultural significance, where we may not be familiar with the oils at all. We may not know the origin, there may not be any documentation. So oils are ubiquitous throughout the museum, some of which we're very familiar with, and some of which we know nothing at all. So a little bit about paint. So paint is a binder and pigment mixed together, and it could be a mineral pigment, like from a rock. It also could be synthesized, and it could be from an organic source, such as insects or even plants. But these are all mixed with a binder to make the paint. So you have protein-based paints, such as animal glue, which is collagen extracted from skins, bones, or tendons. You have casein, which is extracted from milk. And you have whole egg or egg yolk. But you also could have polysaccharides, such as gum arabic, or lipids made from plant oils. And once you grind these things together into a paste of the consistency that you desire, you have a paint. And binders make a difference. So the color is important because it gives you the created image that you see, but the binder affects what you see. So here I'm showing you a range of different binding media with the pigment ultramarine. And you can see that some of the binders saturate the pigment, so it looks darker and richer. And other ones you have more of a lighter appearance because it's scattering the light more. So binders do affect what you see or how you comprehend an image. And artists, especially modern artists, will select the binder to create the effect that they want. And here I'm trying to show you that binders have been used throughout time. So the paintings on the top are much older than the ones on the bottom. But yet artists today will still choose things like distemper, animal glue, or egg tempera to create the image that they want. So it's a very artistic choice, and artists are familiar with materials and properties, and they make this choice. So plant oil binders. The most common plant oil binders in Western Europe are linseed oil, poppy seed oil, and walnut oil. And these are considered drying oils. They dry by auto-oxidation to create a hard film that is very saturated and can be glossy. You can modify these oils. You can heat them, you can add dryers, you can add things to them, and you can change the way they work. They also have some intrinsic properties that make them different. And as you can see, the linseed oil is substantially yellower than the poppy and walnut oil. And artists who use these oils are aware of these limitations. So an artist might choose to use poppy or walnut oil for colors where the yellowing might not be such a great idea, such as in the whites. But artists were aware of the material properties of the oils and how they could manipulate them. And they would take advantage of that to create these beautiful artworks I show you here, where Rembrandt and Velázquez and Rubens all manipulated the oils to create the smooth surfaces they wanted, but also to add the texture. Linseed oil is one of the most popular oils and prevalent, and it is a triglyceride, which I show you on the upper right. So it's basically glycerol with 3 unsaturated fatty acids. Linseed is a very popular oil also in industry, so there's lots of different ways that you can manipulate the oil, again, with heating or additives. Every time you do some sort of a process to linseed oil, you're going to change the chemistry slightly, so you can change the working properties. And if you look at the fatty acid profile of the drying oils, the classic drying oils of linseed, poppy, and walnut, I draw your attention to linoleic and linolenic acid. They're 2 of the unsaturated fatty acids. By definition, high levels of those 2 fatty acids means it's going to dry pretty fast, hence drying oil. But if you have a high concentration of linolenic acid, that's an indication that your oil may yellow over time, especially in the dark. So even though linseed, poppy, and walnut are similar and considered drying oils, there's a slight difference. I put this up not because I'm going to quiz you later, but this is one part of the mechanism of linseed oil. So linseed oil has been extensively studied by chemists to understand the properties of the mechanism of drying. And the reason that so much attention has been put on this is because, as I said, linseed is one of the most popular oils used by painters, and so it impacts our collection. We want to understand why artwork is changing. We want to understand how to best treat it, how to best preserve it, how to best interpret it. So a lot of attention is paid by chemists to molecularly describe the process of drying. I also want to point out that there has been some interest in understanding the role of a mucilage within oils. And there's some discussion about transient species which occur, but in the end, you're going to make a dry oil, and you're going to be following these mechanisms. Chia oil is also considered a drying oil. It's a triethylglyceride. It has the similar fatty acids and slightly different concentrations. But if you notice on the picture on the far left, the raw chia oil, freshly expressed, has sort of a cloudy, turbid appearance. And even when we treat it with heat and dryers and other things, that turbid appearance remains. We've observed that the chia oil actually is kind of slimy compared to linseed oil. And we believe that there is some sort of mucilage fraction that's still inside the oil. And some of the work that we've done in mechanical testing seems to be carrying that out. And Jose showed you a little bit of that in his talk. One of the interests for the working properties has to do with how the paint is made. When you're grinding a paint, if you're going to brush it out, you'll be looking for a particular consistency. Whereas with the lacquerware, you're essentially compressing dry pigment into the oil and then adding the oil and pigment alternately. So they're slightly different approaches, but they're essentially the same type of thing, a pigment and a binder. And if we look at the fatty acid profile of chia compared to linseed, again, you'll see that there's high concentrations of linolenic and linoleic, and the high concentration of linolenic may mean that chia oil has a propensity to yellow. And then we have aje, which is our other source of fatty acids in the lacquerware. And that's a completely different material. It's a solid at room temperature as opposed to a liquid. The high concentration of stearic acid has been quoted as the reason that it's a solid. And it also has other molecules in it. So aje itself has sort of a reddish, orangish appearance, and that's been attributed to compounds called carotenoids, which give you the color. The aje, when I show you there on the left side, the interior of that is still soft. It's kind of like the consistency of peanut butter. And that has a lower melting point than the exterior, which is oxidized over time. And when we were looking at the different recipes, what we noticed is the unoxidized material will melt into the oil more readily than the oxidized material. So you can look at the oils of chia and linseed, because I'm trying to distinguish between these two using light techniques such as Raman or Fourier transform infrared spectroscopy. But those techniques really excel when you have a pure oil and a fresh oil. But once you start to add pigments and then you dry them and you have lots of oxidation and side reactions, it becomes much more difficult to make a distinction between those two. So another way to do this work is to break the molecules down to smaller pieces and use a technique called fatty acid analysis using gas chromatography mass spectrometry. So what we do is we take the oil, whether it's from an object or a liquid, we do something called transesterification, which breaks it into smaller molecules. We can separate it on the column. And then on the lower left, that is a chromatogram of the fatty acid separated out. And then, you're kidding. I'm going to go over. And then on the right is the mass spectrum, which is each of those peaks that's fragmented, and you can identify the molecule that way. So this was a technique that was established by Mills and White in the 70s, and they established guidelines for how to describe oils. And this is for classic drying oils. So again, linseed, walnut, and poppy. We have ratios of fatty acids. Azolytic palmitic defines a drying oil if it's equal to 1. Palmitic to stearic is different for each one. So ratio of 1 to 2 for linseed, 3 for walnut, and 4 to poppy. If we look at the reference oils, which we obtained, you can see that there's sort of a repeated pattern for each of the oils. And the gray columns are the A to P, or azelaic to palmitic. The orange columns are palmitic to stearic. When we look at our paint models, you can see again, they're kind of consistent. But then when we move to objects, we have such a variety of fatty acid ratios that we really can't say what's linseed oil and what's aje anymore. If we look at these objects, and you're going to see these in the next talk, I also did the analysis on this, and what I want you to take away from this slide is not that just that there are differences between each object, but there's also differences within each object. So the fatty acid ratios aren't doing such a great job to help us distinguish between linseed and chia. I also want to point out that with GCMS, you can identify waxes or resins or even pigments that are in the objects. I've also looked at the painting, and again, we have some unsatisfactory fatty acid ratios, but I am able to identify pine resin and collagen glue, but I can't tell you what layer that is in. So now what do we do? We've tried fatty acid analysis, it's not working so well. So what we can do is take a step back and we look at the larger molecules rather than breaking it down to smaller pieces. So now we're going to look at the entire lipid concentration. For this, we need to partner with ARCH, which gives us the instrumentation that we need and also the expertise. And without this being in place, we could not have done this work. So for the lipidomics, we use two techniques. One is called MALDI, matrix-assisted laser desorption ionization, thank you, Alicia, and direct infusion Orbitrap. And I'm only going to show you MALDI today. But the point is we take a single sample and we can extract out the protein fraction and the lipid fraction on a single sample, and then we can analyze them each separately. Today I'm going to show you data that focuses mostly on the triacylglyceride region because that is the largest concentration of molecules within the lipid profile. But we did look at several lipid classes, including phospholipids, sterile esters, fatty acid methyl esters, for both the linseed oil, the chia oil, and the aje reference materials, and also for objects. So here I'm showing you an analysis on liquid oil, of wild chia oil and linseed oil, and you can see that the triacylglycerides pattern is essentially the same for both of them. The intensity is relative, so it's not diagnostic. So you can't say the high peak at 57.7 is indicative of linseed. And we also compared our data to the literature, and there are only two triacylglycerides that vary distinctly between chia and flaxseed oil or linseed oil. But there's so much variation depending on where the material was grown, where the plant was grown, when it was harvested, how it was processed. So again, it's still not diagnostic of chia versus flaxseed. And here are the TAG regions for 5 different chia seeds, chia seed oils, and linseed oil, and poppy and walnut. Poppy and walnut have very distinct profiles. They're actually missing some of the TAGs. But if you look at the linseed and chia seeds, the pattern is very, very similar. But we still have to prove that each one of those peaks is exactly the same. Because you could have 2 molecules that have the same mass to charge ratio, but have a different chemical composition. So the next step is to go through every single peak and do what's called MSMS, where we can actually get the sequence structure of the triacylglyceride. So here, for one of the peaks, we have chia oil, linseed oil, poppy oil, and walnut oil, and they're identical. They're all the same. But then we'll look at another peak, and it's the same for chia oil and linseed oil, but different for poppy and walnut. So everything that we've done the analysis to make sure that we can tell exactly what the composition is, and we have not been able to find something that is distinctive to be able to identify chia oil versus linseed oil. We also applied this to objects, and what I would like you to notice, so this is the lipid, the entire lipid profile. We've been talking about the TAGs, which are on the upper right, and you can see as you move straight down that as you look at objects, those TAGs have either oxidized or broken down into smaller molecules. And again, we've gone through every single one of those peaks, and we're not able to find anything that's consistently different between a chia oil or a linseed oil object. So where do we go from here? So what we decided was to, again, take another step back and appreciate the fact that oils are extracted from seeds. Seeds are biological material and they contain DNA. So especially with chia oil, which we know is rudimentarily processed, if you squeeze out the oil, there's a good chance that you're going to squeeze out something else in there too, possibly some DNA, possibly some protein. The DNA in the seeds is transcribed into proteins, and some of those proteins are enzymes, which give you the carbohydrates and lipids that we've been talking about. The proteins and carbohydrates make the structures of the cells, and that makes the plant. So these molecules are more biological or species specific than the lipids, or could be. So in the next two talks, we'll go into more detail there, so I won't go into that. But I do want to say two closing remarks about where we are. One is this interesting sort of mucilage consistency of the chia oil. We're interested to understand how this component affects the drying, the actual behavior of the drying, and that's the work we're doing with City College, but also the drying mechanism. It may retard the mechanism somewhat, but it'd be interesting to look into this. And the other is, what is the role of aje in how the lacquerware is made, the mechanical making of the lacquerware, but also the impact of the drying mechanism of chia oil. And finally, just to echo what Andrea said at the beginning of the session, is that this philosophy, this protocol that we've put together, could be applied to many materials that are in the world that we don't understand or we have very little information on. The reason, one of the reasons we were very successful was we had a question that was of interest to us, but also interest to others who already had been studying this, looking at it, having questions. So by collaborating the Met and its resources, our ability to do very complicated science with people who had an interest to see this succeed and that we were helping them to answer a question, that is what made this successful. We hope that we can apply this to other things. So if you have biological materials in your collections that you're interested in, we'd love to talk to you about it. And with that, I will close.
Thank you, Julie. So our next talk, which I will try and pull up here for you guys, is, or you can do that while I do this. Our next talk is going to be co-delivered by Alex Popowich and Sarah Knoll. I'll introduce Alex first. Alex Popowich is a conservation scientist at the Philadelphia Museum of Art and a member of ARCH. Alex was a research associate and postdoctoral fellow in the Met's Department of Scientific Research from 2020 through 2023. While she uses both mass spectrometry and spectroscopy techniques to characterize cultural heritage, her specialization is studying the artistic uses of proteins and lipids by mass spectrometry. Previously, she was a postdoctoral fellow at the Smithsonian's Museum Conservation Institute, focusing on the characterization of decorative metal threads and the development of gilding technology from medieval to contemporary times. She holds a PhD in chemistry from the University of Alberta and a bachelor's in chemistry from the University of British Columbia. Alex has also assisted the conservation departments of the Royal Alberta Museum. So Alex first. So our next speaker, I'll also introduce Dr. Sarah Knoll. Dr. Sarah Knoll is the visiting assistant professor of conservation science at the Conservation Center of the Institute of Fine Arts at NYU. She was previously a research associate in the Department of Scientific Research at the Met, focusing on the use of proteomics to classify trace proteins in lacquer and paint. Sarah holds dual BAs in chemistry and German studies from Pomona College in Claremont, California, and a master's in chemistry from the University of Cambridge. She received her PhD in chemistry from Stanford University, and prior to joining the Met, Sarah has also taught general, analytical, and physical chemistry at Pomona College. Her research interests include the development of new methodologies and novel applications of high-resolution mass spectrometry for bioorganic molecules with a particular interest in art materials. She is also a member of ARCH. So I'll turn this over to them.
Thank you. Let's like center it.
Hi. Hi, everyone. Welcome to the Protein Talk. Alex and I are really excited to present some of our work and to show you a new method for protein identification in dried oil paints and lacquers. And studying proteins in seed oils hasn't really been done before, so we're actually quite excited about the potential for this work when it comes to binder identification. And
You ready?
yeah.
Okay. So Julie set us up a little bit with our somewhat unsuccessful attempts with lipids. So as she said, we decided to look at proteins, and proteins in oil, kind of a weird idea. There really aren't much there. So in order to do this, we kind of needed to do two things. The first is to build databases, so protein databases for the species that we're looking at. So we're going to be talking about both chia and ahe as we go along. And also validate that our methods work. So we do this, and I was trying to see if I could get a pointer. Wait, you see it? Yeah. Okay, perfect. I don't know. Anyways, so starting with the material with the most amount of protein. So that's the seed itself going up through the processing. So the toasted seed into the oil before we get to the artwork. Our end question is painting. So where we don't know if chia oil was used at all. But we can use another art form, the lacquerware, where chia oil theoretically is used in specific regions as a kind of type of reference material to validate our methods and see if they are working on both contemporary and historic objects. And then kind of as we go with decreasing protein content, there's a lot of other unknowns. So, there's mixtures, could be mixtures of oils. There's a lot of other proteins that are going to be in higher concentration in the samples that we're looking at, especially from conservation, human contamination, other binders in there, degradation, and so forth. Here are some of the reference materials that we did use, both from the Met and sourced from Mexico. I'm just going to point out a couple of them. So we looked at two different variants of chia seed. So the black chia seed, which is cultivated in the US, and then this Guerrero variant, which was wild and picked in the state of Guerrero in Mexico. And then we're going to be talking about two different types of oil, as Jose had touched on. So one made in the more traditional method where the seeds are toasted, ground, then wetted and pressed, and then one where we made it with the machine. The reason for this is that we needed a larger amount of oil to make our mock-ups and for other studies. And as you can see, our yield from the traditional method was very small. We weren't that strong. So we wanted to really validate that the protein content for our purpose of the studies was the same. So we did go through, and we'll demonstrate that for the purpose of the talk that we're giving, they were similar enough.
So we used what's called proteomics to do this work. And very generally, proteomics is a study of all the proteins in the sample. And it's not just identifying the proteins, it's looking at how they interact, how they might be modified, how they could be degraded. And we're doing this with mass spectrometry, and in fact, a very high resolution mass spectrometer that allows us to very precisely measure the mass of ions, and this enables our identifications. But of course, to actually analyze the protein, we first have to get it out of the sample, out of the sample matrix, whether or not that's something like a painting or a lacquerware object, or in fact, liquid oil. So once we get it out, we are doing this with a filter-aided extraction, and we have the protein. We then use an enzyme to effectively chop this protein up into peptides. Those peptides we are then separating on an LC, and we analyze on a mass spectrometer. In fact, that mass spectrometer actually fragments this peptide further into what you see as this MS2, and that enables our sequence identifications. Now, just to kind of remind everybody, a peptide is just a segment of a protein. So once we have these shorter sequences, we're able to piece them together a little bit like puzzle pieces to form an overall picture of the protein. And this method is not biased towards Chia, and I think Alex has really indicated this. We are pulling out any protein that is present in the sample, whether or not it came originally from the object, from a conservation treatment, or in fact even from handling. And then one other thing, and Julie alluded to this, it's actually possible to do the lipidomics method that she talked about and this proteomic method on the same sample. And so it's really valuable that we can get all of this information from one piece.
Okay, bear with me. This is more just to give an overview of what comes next for scientists and to give a perspective of kind of the time that this takes. So once we get that raw data, the mass spec data, which we call MS2, so we fragment the peptides, we get spectra. Then we go through a process called bioinformatics. So it's a heavy computational process of actually taking that data and identifying what it means. So first we do refinement to the data. So we kind of deconvolute the peaks, we normalize their intensity, we align them. Then we can sequence that data to actually identify like what amino acids the peptides are in two different ways. One is called de novo sequencing. So based on the different masses of the peaks, we can identify what amino acids are in that small peptide just using that data alone. The other is doing database searches. So we take either publicly available databases or databases that we create using custom sequences that we make, and we compare our data to that to do searches. So we're going to be talking about these two different types of sequencing. So in the case of AHEI, that L-axinaxin insect, for example, it's never been studied before. So we'll talk about de novo sequencing. Chia has been sequenced, so we'll do a lot of database searches for that. After that, we can look for peptide mutations. So this is looking for one single amino acid that has changed either between species or for a specific individual or something. So we'll give an example. When we're looking between the black and the Guerrero variants, we can actually see within a single species, there's differences in the proteins. Now, at this stage in our bioinformatics, we can both do protein assignment, so identify all the proteins in our sample, and identify the species. And as we've said, again, this is very comprehensive. So any proteins that we extract, again, not just like plant, it's whatever database you're searching. So Mammalia, you can look for collagen glues, human contamination, egg proteins, casein, so forth. We can identify what's there. But it involves a lot of interpretation and manual confirmation, and unfortunately for us is really time intensive. But we use a alignment algorithm called BLAST, which some people might know or hear us mention, to check for species specificity. We have to check all of our raw data to see if the fragmentation is complete, there's enough intensity, what the scores are, if there's alternate IDs, just to really confirm that we do have it in our samples. And then after this, you can do additional bioinformatics. So you can look for post-translational modifications. These are things such as signs of aging, so oxidation or deamidation. You can look for tissue specificity. So these are things, we'll mention a lot of seed storage proteins. These come from the embryo of seeds, so that's one example. There's a lot of research done with mammals, which makes that really effective. Then quantification, we'll give example of some quantitative data, and then even further looking at protein interaction networks. Sorry, heavy, that's it.
Okay, so the question is, we've got this method, is it actually going to work? Are we going to be able to pull out protein from the oil? And the short answer is yes. And what you're looking at here is the quantity of protein and peptides that we can pull out from a sample that's just been prepared for proteomics. Those are the ones labeled EFASP, and a sample that's been prepared for both lipidomics and proteomics. And those are labeled delipidated. And what you can see is we do have a little drop in numbers as we come down to what is a more relevant and realistic sample size. But we're also able to pull out protein from semi-cured films, so paints that have started to cross-link, which is very important, of course, as we're thinking forward to objects. And then we're also able to pull out protein from oils where we actually don't quite know how they were processed. So something like a commercial oil or an oil that was purchased at a market. Now, what we're looking at here with these numbers are actually plant proteins, and unsurprisingly, what we really care about are chia proteins. So this heat map is our kind of semi-quantitative data, and what it's showing you is the coverage of representative chia proteins across these different classes, and then across our full kind of reference series. So from the seeds in that top row through various fresh oils to dried oil films, and then at the bottom into paints. And I think two important things to point out on this slide, besides the obvious excitement that we are seeing chia proteins. The first, as Alex mentioned, when we use this traditional method to prepare oil here at the Met, the volume that we obtained was quite small. So we wanted to verify that the protein coverage we saw in the machined oil that we made was similar. And so you can see the kind of machined and traditional in these top three rows. And this confirmed for us that the protein kind of coverage looked quite similar and gave us some confidence in using that machined oil to prepare our model samples. The other thing to point out is when you're kind of looking down, it's really the seed storage proteins, it's that kind of leftmost class, that we see the best coverage all the way down through the paints. And we'll in fact use seed storage proteins when it comes to species-specific identification. And it also turns out to be not so surprising that we see the best coverage when it comes to that class of proteins. So a chia seed is about 20% protein, and of that protein, 95% are seed storage proteins. And these, as Alex mentioned, they're found in the embryo. They provide a reservoir of amino acids for when the seed is germinated and then starts to grow.
So before I jump into the next topic, I just want to say we're excited by the last data because that's the first time that anyone has ever looked at proteins in seed oils. So doing that was like exciting, especially at like 10 microliters and like on commercial oil, like in any industry, food science, anything. So yay. Okay, so I did mention we looked at two different variants of chia seeds. So these are within the same species, and there are actually different types of chia seeds. They're all Salvia hispanica, the same species. We looked at a cultivated variety, the black, which is the type you most commonly eat, and this Guerrero, which we had from wildly grown in the state of Guerrero in Mexico. And we were interested to see if we could see any difference in the proteins. And what was interesting is that within the seed storage proteins, which we said are the main components, we actually could. So here I'm showing the 11S globulin, which is this protein, and all of the gray is the coverage that we got on the black chia seed. And these are the MS2 for 2 peptides of the black. But when we look at the Guerrero, we actually have 2 point substitutions for amino acids that are different within the same species, which isn't actually that common. So that was really exciting. And something that we thought, okay, this could inform possibly on what type of seeds were being used in artwork. Now, you'll see later that when it comes to artwork, we don't get this kind of coverage. Like this is from a seed itself, not even, necessarily the oil. And we also didn't study all of the variants of chia seeds. So we couldn't say, oh, it's Guerrero, oh, it's black. And there's a lot of genetic variation that occurs over time. So even these seeds that we're studying today are not the same necessarily mutations that would have existed in the 1700s, but it is informative information. When we did look at our paint mock-ups, we could see what type of oil seeds were used, which was exciting. Just to touch back to the ahe, so we looked at the proteins that we could find in the fat from this insect as well, to see what sort of information we could pull out. This isn't the full list, and this is a homology search against the database for insects, for true bugs. We also did de novo sequencing, so sequencing the peptides, the proteins themselves, without databases. But just to point out that we're seeing highlighted in yellow a lot of muscle proteins, heat shock proteins you'll see mentioned, which are stress response, and then structural proteins. The issue of these types of proteins that are left in the fat is they're very well conserved between species, even between bugs and mammalia. So they're not that species specific. So you'll see later on that when it comes to the ahe fat, we can get to a genus, but it's more difficult to get to the species. So there are other techniques, such as in the next talk, the work that's coming on or being done with genomics, that may offer a better level of species specificity than proteomics for that material.
So having had success with the reference materials, we then of course are looking at objects. And objects come with their own unique set of challenges, and quite a few challenges. The first is the sample size, I think, as everyone knows, is extremely limited. And if we were looking at something with a proteinaceous binder, we might expect that the protein content is around 10% by weight, but we're interested in things with an oil-based binder, and the protein content we're expecting to be far less than 0.1%. So we're really coming up against very trace levels of these proteins from the oil. And then I think a theme that is emerging, we're not just seeing the proteins from the oil, any other protein that is present, whether or not it might be a glue in a ground, a wheat starch protein from a lining, a human protein from handling. These are all things that we will see and we have to kind of tease apart. And the databases. Our data is actually only as good as the reference databases. And this is something that's continuously evolving. So we might have a peptide that when we search, it looks like it's species specific today. It doesn't match any other species in the world. But actually what that really means is it doesn't match any other protein in the database on, today, October 15th. Mm-hmm. But 3 months from now, someone could upload a protein and it might match that. But we also, of course, then have species that are not in the databases that we need to sequence and create our own. And then the last thing, and I think Julie mentioned this in her talk, in order to do this type of research, we need access to the instrumentation, and these high-resolution mass spectrometers are typically found at academic institutions. So having a partnership like ARCH is really important for enabling this work. So given that there's some extra challenges, we wanted to give ourselves the best chance of success. So we also developed what is called a targeted MS method for chia proteins in the objects. And for any mass spec people in the audience, this is a data-dependent acquisition with an inclusion list. I know there are a few of you in here. But what this is in a more general sense is we chose 3 of these seed storage proteins from our reference seed. And we picked ones that had high expression, high coverage. Actually, if anyone saw the Nobel Prize in Chemistry was awarded for predicting protein structure. You can see the predicted structures using this on the bottom. Thanks to Alex for actually putting those in. And so these are our 3 proteins. We went through all of the peptides from those proteins and picked out those that were specific to either Salvia hispanica and some that were actually specific to the genus Salvia. And then that gave us about 650. We sorted these by retention time and then their mass to charge. And then when it comes to the experiment, the instrument uses this list to actually search specifically for these peptides. And this is important because the instrument is otherwise kind of basing its choices off of intensity. And as I've mentioned at the beginning, we're expecting these oil proteins to be very low level, therefore low intensity. And so this helps to make sure that we're not missing them. So using this, we've now looked at 4 historic and 1 contemporary object, and Alex is going to talk about the first, this casket.
All right, so I'm going to kind of fly through because they're tables that aren't too exciting, but this is sort of what our final data looks like. So from this casket at the Hispanic Society, we get a list of different seed storage proteins. As you can see, their names, mostly 11S globulins on the far left that we're detecting, and some of the peptides. In the far right column, it kind of lists whether they're unique or not to the different salvia species. What we do see is that we have one little peptide that is unique to Salvia hispanica. And when we go back and look at the raw data, so the MS2, we can say, okay, it's good quality, good intensity, fragmentation, and the amino acid that's unique to that is there. Everything's good. So we do know that it is chia oil used in this. Another example, as I mentioned with the ahe, it looks like we have a lot better coverage, a lot more peptides, but this is a muscle protein, so a lot of those aren't actually unique to insects, but some of them are. So we can say, okay, it falls within the true bug genus, but we can't get to the species for this chest here.
And then this is the third object, this blue desk from the Museo Amparo. And this is another seed storage protein, another 11S globulin, again showing our species-specific peptides that have that very nice fragmentation. But you'll notice the species is no longer chia. It is, in fact, flax. So we found actually quite a few proteins within this sample that corresponded to flax, or therefore likely linseed oil. And again, we have in this final column here showing how many of those peptides were actually species specific, which of course gives us greater confidence in making this assignment. And for us, this was actually a really important control because we wanted to make sure that our method, again, was not biased to chia, that it in fact could identify linseed oil if it was there. We weren't necessarily expecting it, but we did in fact verify that. And so looking at all of the objects kind of overall, as mentioned, we've now found chia within the casket from the Hispanic Society, a dowry chest here in the Metz collection, of course, the contemporary gourd, and then linseed oil in this blue desk, and then as Alex has mentioned, these bug proteins within the paraben chest. Now, you'll notice there's asterisks on quite a few of these, and I want to kind of point that out specifically. That's because these assignments come from one sample site on each of those objects. And so, say, take the blue desk, for example. Although we found linseed oil in one sample from one site, we're actually actively analyzing data from other sample sites. It doesn't mean that we will necessarily find linseed oil elsewhere, or that we might not find another oil used in another part of this object. So just a kind of a reminder on the context there.
So I'm just going to give a quick example of data in paintings, and we won't get to like the end of the story with this one, but just to show what we can get. So we've mentioned this Jose Sanchez painting a few times. So the sort of things that we do see, like I said, we can see the collagen glue. So we know that a cow glue was used here. We also can say, you know, there's a wheat paste lining, so we see proteins from common wheat. But in this one, like we said, it gets more complicated because these proteins are really overwhelming the other plant proteins. And for this one, we suspect there's maybe some other things. We're working on the data going on. And limitations with the database. So with that, we'll come to our conclusions and kind of point out a few things that are both good and challenging about the field of proteomics. So as a whole, proteomics can really provide a comprehensive view of the proteins in a sample, even at trace levels. And we are able to identify seed proteins from drying oils. And this is the first time that any proteins from seed oils have been identified in any field. But we know that cultural heritage samples are incredibly complex due to aging, chemical interactions, and contamination. And so one of our major limitations really comes to the databases. So as Sarah touched on, our results are only as good as our databases. And with a lot of the samples we've worked on, we really have hints that there may be other plants or other species used as materials in these objects, but the databases for plants, insects, and other materials really aren't complete. And so we have to ask ourselves, okay, are we going to spend time sequencing all these species, or are we going to come up with targeted questions like with the chia? And ourselves, we don't really have time to be sequencing all these proteins at this time, but it kind of is like giving you ideas of things that might be out there that we're not seeing. But something that is good is that we can always reprocess our data as the databases expand and new materials are released, And one example is that, like a month ago, there was a new salvia species released, and we're like, recheck our data, make sure everything's specific.
And there are other techniques, I should say, beyond proteomics that can offer more species specificity, something like genomics to tee up the next talk. And this can become, I think, really important for cases like AHE, this bug, where these proteins that we're seeing actually are very similar to ones that are in mammals, that are in humans, and you almost need something that's even a little bit better to try and tease those apart. And then just a reminder, I feel like we're maybe a little of a broken record here, but proteomics really can be used to study any protein. And so, of course, in the context of this project, we're interested in chia, but I think as Alex has alluded to, we are seeing so much more. So even I think as you're thinking of your own objects and the own things that you might be working on, any protein from anything we could possibly see. So I think that's one of the really exciting things about this work is it actually isn't specific to chia. It has so much more potential than that. And thank you, I should say, huge team to make this happen. Many listed here, and then of course to the Museo Amparo, the Hispanic Society, and the Met for opening their collections. Thank you.
Okay, thank you, Alex and Sarah. So we're going to move on to our last presentation for the morning. Jeremy Wayne Hirschberg. So Jeremy Wayne Hirschberg is a biologist in the Mason Lab at Weill Cornell Medicine, conducting genomic and metagenomic research for space missions. He also helped found the world's first aerospace medicine biobank, a repository of human and environmental samples from space missions, which aims to increase the accessibility of samples for space medicine research. Previously, Jeremy worked in diagnostics, developing diagnostic platforms for COVID-19, influenza, and other biological targets. Now he aims to apply a biotechnological approach to address the challenges of the future and sending humans to space and answer questions about the past and analyzing historically significant artifacts. So I'll open the floor to Jeremy.
All right. Hi, everybody. Thank you for the introduction. I'll be presenting today on genomics for the characterization of artistic materials. And this is the DNA talk. So I'll just jump right in. So I work at the Christopher Mason Lab at Weill Cornell Medicine, and our core competency is and our interest is to study DNA from interesting places. And what that means is many different things. So for example, our Two Frontiers project looks at the DNA of organisms that live in deep sea volcanic vents. Or the Metasub organization looks at the DNA of organisms that live on the New York City subway, along with transit cities all around the world. And we also look at how DNA is affected by going to space environments. And so it was a natural partnership with the Met, and we're grateful for the opportunity to look at the DNA that we might find in objects of cultural significance as well. So let's start with the big picture. What is genomics? So at the foundation of any biological organism, we have the molecule of DNA, deoxyribonucleic acid. That's this double helix ladder structure where the rungs are made of base pairs, A, T, G, and C. A and T are always together, G and C are always together. And this creates a code that ultimately codes for cellular function and on a complex level, biological life. So in any organism, you have this molecule and it gets wound up and wound up and balled up and balled up into chromosomes. And chromosomes are stored within the nucleus of any cell. And it can vary between different species of varying complexity, where bacteria often only have a single chromosome, where we have 23 pairs or 46 chromosome. And there's some plant species that have hundreds of chromosomes. And when we talk about the genetic information, all of the A, T, Gs, and Cs that comprise the entire chromosomal complex of an organism, that is what we refer to as the genome, the whole big picture of genetic information for that organism. So how does genomics differ from genetics, which might be a term people are more familiarized with? So genetics studies the way that certain traits arise from particular regions of the genome called genes. So we think of heritable traits like eye color might be something that a particular region of the genome codes for. It also, you'll also have genes for more fundamental aspects of biological function. Say, for example, there's a gene that codes for the protein in blood that carries oxygen. So any organism that relies on a blood to deliver oxygen throughout the body will have a gene that produces hemoglobin. Then we also have a set of genes within our genome that are unique to us as human beings, whether it's contributing to our bipedalism or our slightly larger brains. There's a subset of genes that are unique to us being human, and that's where we get into the realm of genomics, where we're looking at, if we're comparing the full big picture of genetic information across organisms, what is conserved and what is different. And so in the instance of humans and chimpanzees, almost all of it is conserved. Those are those fundamental aspects of biological function. And it's only a slight percentage that is really what differentiates us. And as we start to study and learn about exactly what differentiates different organisms, we can do some pretty interesting and cool things with those differences. So that brings us into the realm of chia, and our research question being, can we demonstrate the usage of chia in the making of these culturally significant objects by utilizing genomic techniques to identify the presence of chia-specific DNA that is only housed within chia organisms? So to dive into a standard genomics workflow that I'll do regularly within my lab is we take a sample and extract the DNA out of that sample. This is often done in high school labs with a strawberry or onions, but in our case, we have chia. We pull out the DNA, and then we do what's called a library preparation, which is a fancy way of saying we prepare the DNA such that a next generation sequencer can read it. And typically, going into a library preparation, we need about a microgram of DNA, which for chia corresponds to a couple thousand cells worth of DNA. And we prepare them predominantly by putting these caps on the end that the sequencer can recognize and motor proteins that help move the molecule through the sequencer. So without getting too into the weeds, this is an example of what next generation sequencing looks like, specifically nanopore sequencing. There's a membrane within the sequencer with this big purple pore in it called the nanopore. And the caps that we put on the ends of the DNA molecules can interact with that pore. And then the motor proteins that we've added in as part of our preparation will split the DNA in half and feed one half through the pore. And there's an electrode that spans the pore, and as the DNA passes through the pore, whether it's an A, T, G, or C, will change the electrical potential of that electrode in a way that a computer can interpret as A, T, G, or C, and essentially read that strand of DNA as it moves through. You might be thinking, well, that's only half of the DNA, but because A and T are always together and G and C are always together, by reading one half, we know what the other half looks like. And so when we extract the DNA, it actually gets broken up into pieces. And so as each piece of DNA comes through, we call that a read. And a read can be, for any sequencing run, you can have tens of millions of reads, and they can be tens of thousands of base pairs long. So any given sequencing run will result in billions of A, T, Gs, and Cs. It's a lot of data. And it's so much data that a human wouldn't be able to interpret it in any particular way. But we can leverage high-powered computing to look at these reads and see where they overlap. So if you remember, the genome is present in every single cell, and we used thousands of cells for this preparation. So we actually have different copies of the same portion of the genome that are broken in different places. And so a computer can go in and look at all of these pieces and put it together like a puzzle. And what you get on the other end of that is a pieced together entire genome that corresponds to that organism that you started with. To put this into context and why we call it next generation sequencing, the Human Genome Project culminated in 2003. It took about 13 years, $3 billion, the work of about 20 different universities using devices the size of large refrigerators. To sequence one human genome. Now, with next generation sequencing, I can sequence the entire human genome using a device that fits in my pocket for less than $1,000 in less than a week. And that enables all of these genomic workflows to be possible because of that high throughput and high powered computing that allows us to analyze this data. So once we have a completed genome, we can do a number of things with it. One, we can compare it to other samples to find those differences between different species. We can also publish it if it has never been sequenced before and just say, hey, here's the Chia genome, if anybody wants to compare to it. Or we can compare it to data that's already published. In order to see what we might be working with at that given time. So the summer before I got involved with this project, a Chia genome was published. And so there is a reference genome for Chia that's on the internet. And so this enables what would be the ideal scenario workflow for a genomics lab, which would be to take a piece of a culturally significant object, pull out the DNA from it, sequence it, and then compare the DNA that we got to the published Chia genome. And if they match, we could say, yes, there's Chia present in this object. The problem that we run into, though, however, is we have extreme sample limitations when it comes to working in this type of environment. So these are mock-up paint samples that we're using to refine our methodology before going to a very important object. But that small black dot in the tip of my finger corresponds to the approximate sample size we might be able to expect to get from a museum object. And the thing about that is a sample of indigo paint, which is only 64% Chia oil. And plant oils typically do not have a lot of cells in them. As Julie mentioned, we expect that in the making of the oil, some cell debris has made it in there. And so we expect to find some amount of DNA. But when we talk about an ideal sequencing run requiring thousands of cells, the chances of us being able to get that amount of DNA from a sample like this is fairly low. So, as unfortunate as that is, we have another tool in our toolbox that we can utilize, namely PCR, polymerase chain reaction, which is the exponential copying of DNA molecules. Some of you may have become familiar with this technology in recent years, unfortunately due to the COVID-19 pandemic. But this nifty piece of biotechnology revolutionized many fields of biological studies, including clinical diagnostics, forensic science, genetic testing, and it was so impactful that it was awarded the Nobel Prize in 1993 in chemistry. And so to go into a little bit about what PCR is, it was discovered in studying the way in which cells replicate their DNA. So as any biological organism grows, its cells have to replicate. And when they replicate, they also have to replicate their DNA so that every cell has a copy of that genome, the instruction book for what that cell needs to do. And so it was observed that during this DNA replication, there was a molecule called DNA polymerase that was identified as performing a particularly useful task, in that if there is a single strand of DNA that is just half of the molecule, DNA polymerase will go in and move along it and pull, and as long as there's free floating AT, Gs, and Cs in the solution, it'll pull down the corresponding base as it moves along it and build out the second strand of the DNA. And it'll do this entirely on its own. The only things that it needs are free floating AT, Gs, and Cs and an anchor point to start at. And so someone cleverly found out that if we take a strand of DNA on its own and we heat it up, it'll split in half. So now you have that single strand, and we can put in these little orange, or the red arrow, orange region on the side, are little anchor points that we can put in there that we call primers. And once you have that single strand of DNA, AT, Gs, and Cs floating around, and your primer, DNA polymerase will go in there and build out the remainder of that strand. And so because A's and T's and G's and C's always go together, when you have one strand and you split it in two and build out the rest of that, you're essentially left with two strands in the end that correspond exactly to the starting strand that you started with. Then, if you heat that up again, both of those strands split in half. You start the process over, the anchors come in, polymerase moves across, and you go to 4 strands, and then 8, and then 16, and before you know it, there are millions and millions of copies of this same strand of DNA. And this is why it's such a revolutionary technology, because you go from a single strand of DNA that's not detectable and not useful to millions of copies that you can easily detect and use for different things. Another nifty thing about PCR is we can design these anchor points, the primers that we have, to target a very specific region of DNA. So this is how COVID tests work. If you think, for example, you do a nasal swab, your nasal swab is going to be filled with all sorts of genetic material, a lot of human genetic material, other microbes that live in your nasal cavities. And if we made millions of copies of all of that, the picture would get kind of muddled. So what we can do is, if we sequence the COVID genome, we know what region of that is specific just to COVID. And so we can design these anchor points to go in and only bind to that region of DNA, which means if you have a sample and there's no COVID present, no anchor points bind, DNA polymerase has nowhere to build, and you don't get copies of DNA. So when you have a COVID test, this would be an example of several positive COVID tests. This is an amplification plot, the type of data we see from PCR runs. The only time you see this massive spike in signal is when DNA replication took place. So if you went in and you had a COVID infection, the first cycles that you see there, that's your COVID, whatever amount of COVID DNA you had inside you is going from 2 to 4 to 8 to 16. It's not measurable. But then by the time it's up to hundreds and thousands and millions, you get this massive spike. And the loading of DNA that you start with will determine how late or how early you get a positive signal. So that's how COVID tests work, and we can actually do this exact same thing for CHIA. So because that CHIA genome was published, we can go in and identify CHIA-specific regions that aren't shared by other species and design these anchor points to go in and find and bind to it. And so that's what we did. These are amplification plots that we've generated using this CHIA test. And so we validated the test using DNA pulled out of chia seeds. So we know it's CHIA DNA, and we tested just how little DNA we would need in order to detect it, which we found to be a single cell of CHIA theoretically is all we need to have present in order to get a positive result from our chia test. We also did specificity testing. We want to make sure that if there's something else in there, just by chance, our anchors don't accidentally bind to that. So we tested it against ahe, flax, human contamination, and that's a positive control to show that the assay actually ran properly, but nothing else amplified, which means if we see a signal, we know that chia is present. And if we don't, we can't necessarily 100% say that it's not present, but we have a pretty good indication considering how sensitive this assay is. So the next step is to take our mock-up paints as a practice, extract DNA from them, and even if we can't measure any DNA that we've extracted, as long as there's a single cell's worth, we should be able to amplify it and see a signal. And we have seen that. This little blip at the end, and it's small because there was only a little DNA present, shows a positive PCR result to detect chia in our mock-up paints, which we knew were made with chia. And this is kind of where we stand now. The problem is we're only seeing amplification in about a quarter of the replicates that we're testing. And we have really good indication, because we know how sensitive this test is, that the bottleneck as to why it's not all of our replicates comes to how efficient we are at pulling the DNA out of the paint. So it's not that there isn't chia in there. We know it was used to make it. It's how good are we able to get that DNA out of the paint sample. So we now have colleagues at the Museum of Natural History who are well-versed at pulling DNA out of difficult samples. They do it out of fossils, optimizing this extraction process to increase the amount of DNA that we get out of these paint samples. And while we haven't run the PCR yet, we have reason to believe that they've increased our DNA yield, and we expect to see the PCR results to follow. And once they do, then we can take the next step and actually test this on an actual object. So that's where we stand right now for the chia test itself. But I wanted to quickly mention as I wrap up just how widely applicable this is to other biomaterials. So if we take an organism like the L-axanaxin, which is responsible for making this the waxy substance used in lacquerware. As Alex mentioned, it's not widely studied. There's no reference genome. If we get samples of that bug, we can have plenty of cells to extract DNA from, sequence it, then search through the genome for a ahe specific region, design our anchor points, and have a test that we can validate. Even easier, there are other organisms, such as flax, that are studied in other industries that do have a reference genome already published, which means all we have to do is go into the database, search it for a flax-specific region, design our anchors, validate our test, and now we have a flax test. And what's really cool, I think, about the future-facing aspect of this technology is you can multiplex PCR, which means that it might take a little bit of optimization, but you could run all of these at the same time on the same sample, which means when we're worried about taking little flecks of paint off of these very important objects, you could take one fleck of paint and have a panel of different things that you want to run all at the same time, be that chia, flax, ahe, walnut, poppy, etc. And I think that that's a remarkably exciting future for this kind of technology. And so with that, I'll wrap up. Thank you to the Met and everybody for allowing us to participate in this research, and we're excited for the next steps to come.
Okay, thank you, Jeremy. So let's have a round of applause for all of this morning's speakers. I would like to open the floor for Q&A, so I'll invite our speakers up to the front, get one of the director's chairs and the mic. Okay, so do we have any questions on the floor? Yes, we have one, Monica, in the corner here.
So,
do we have a mic?
thank you, Sarah.
Thanks, Sarah. So first things first, I'd like a special round of applause for Alex and Sarah for being the first to extract protein from seed oils, because I don't feel that first and foremost. And secondly, I have a question for the two of you. It's a bit object specific, so sorry. So I forgive you for not remembering. The first is in
Monica, sorry.
Sorry, is that better?
Yes.
Sorry. So in the object from Museo Amparo, which had linseed oil in it, right? Can we remember where on the object we took the sample?
That one?
Here.
Is it on?
Yeah.
I think it's the lower left kind of panel towards the back, maybe on the
So it was on
Bombay.
the main part of the desk.
But or maybe the Bombay.
Right.
I'm never sure how to pronounce that quite right.
I took the sample, I should
Yeah.
remember. I wanted, that's first question. So, okay, so it's on the main part, because I was, if it could have been linseed because it's restoration
Yeah.
and that's,
Yeah, so one of our suspicions, because we won't go too much into other data we see just yet because anyways, but one of our suspicions with that, because of the quality of the data, is that it is a retouching, because what we do see in the other data is something else. And I'll pass that to you.
I think in some of the other sample sites, we are seeing maybe some peptides indicative of chia, but the quality isn't great.
right.
So I think we're still looking at that and are kind of hesitant to say anything for sure without being a little bit more careful, because they're not quite as good as maybe we saw on some of the other objects. So I think that's kind of maybe why we're being a little bit cautionary and saying, okay, we've seen this clearly in one spot, but I think there's a little bit more work to be done to kind of piece together what's happening overall on this object and kind of to put it into a little bit of a bigger picture.
And so this next question, again, is another anomaly in your tabulation at the end, which is in the chest at the Hispanic Society, S56, you saw bugs, but no oil of any description.
No, I just didn't mention the oil for that
Okay.
one.
And that oil was
So for that one is one of the ones that we're seeing
you can come
we'll
back.
go into it later.
Go into
So
it later.
in some of the objects, we see indication of different materials possibly used, and that is one of them.
Okay.
But I don't see chia in that object.
Or linseed.
Or linseed, no. That's all I'll say. And it's a limitation. So what I do see is something that is not in the database other than very limited amounts. And so I'm not confident to say it's something there unless the proteome is released to a greater extent or we were to develop the database ourselves, if that makes sense. Okay.
Is it on? This is a question for Jeremy and Sarah and Alex. Jeremy, can you take the protein or the data from the protein, from their analysis, and put it into your analysis? It's a double-edged sword question.
Yeah, it's a great question. And in this day and age, we talk about multi-omic research in that I'm looking at genomics, they're looking at proteomics, and really these things, the study of these kind of exist within their individual vacuum currently, but as we continue to build the bigger picture of these databases, fundamentally on a biological level, the genome codes for the proteins. And so inevitably, there will be this interconnection between these fields of studies. However, like proteomics is a fairly new realm of study, and it'll take a little bit of time to really, I think, bring those data sets together.
And sorry, I'll add to that. So it doesn't usually feed proteins to DNA, but a lot, and I would say the majority of the protein databases are actually translated from DNA databases. So the chia database that we're working on, it's not made from mass spectrometry protein data. It's translated from the genome. And a lot of times we have to actually verify from chia seeds reference from mass spectrometry that there's no error in that computer translation, because there can be error when you're taking, a genome database, just translating it to the protein. So they'll publish kind of the genome. Then a few months later, someone will like go on the computer, translate it, remove specific regions because parts of the DNA are chopped out, and then say, oh, here's the proteins now too. But, you know, we have to double check with the mass spec because sometimes there'll be errors in that. But that's usually how the proteins are released.
Well, it makes a double-blind experiment, doesn't it? And the second question, this is a material culture question. Has the provenance of each of these objects been examined? If you have a secretary in the Museo Amparo, and we know that this workshop is working for the vice-regal court, that object might have gone back to Spain. Do you see where I'm going? I don't know that they did in this case. I'm just saying, have we looked at that? Because back in Spain, a retouch would have made linseed oil. Do you see the problem? And we get this with works of art all the time. Where has it been? What have they done? Glue line, paste line. Do you see what I'm saying? Wheat, one case, wax in another. All that comes in, and I wonder how much provenance research is being fed into this hopper.
Thank you, Marcus, for that question. When we went to Mexico and collaborated with the different museums there, you know, we made our best effort to get as much information as we could for the provenance and, you know, the movement of these pieces. For the Amparo desk in particular, we were able to actually use the, you know, really based on observation of the layers. And what's interesting there is that there is a very even layer of what we would call fondeo, of blue paint. It's what appears very much like lacquerware, and then on top a brushwork of, you know, decorations where you can actually see the trajectory of the brush, which is very slightly different to what we see in our batea, right, and your desk, where the background is very nicely polished. You can almost see, you know, it's almost reflective. And then you have the skeins of Pastiglia-like bowl that are applied on the surface. It's hard to say, you know, De la Cerda could have different hands working at his workshop, and I'm not ready to say it is or it isn't De la Cerda, but yeah, we just really using those objects, you know, trying to complement our observations with the provenance that we were able to gather from those institutions, which at times, as you know, sometimes are non-existent.
And I'll add too that when we selected sample sites, we did our best to select regions that we thought were untouched or not retouched.
We have another question in the back. And while the mic is moving, I do have a question for Jose Luis and Julie. So I know that one of the underpins of your work has been working with source communities and local artisans. And I want to ask you, how do you feel that relationship has helped to move the science forward, why it's been so important to you, and how others could replicate it in other places?
Well, that has been key. And, you know, the generosity of people that we visited in Mexico really has been what's made us understand the processes, right? Because we can look at many lacquerware pieces here in New York, in Spain, somewhere else, but seeing it being made, it really changed our perspective as to what to look for. Even formulating our questions, right? Like that's the first time that we really start looking at the question of the mucilage. And what role does it play? Is chia oil an oil that, you know, may be much more akin to an emulsion than a pure oil? You know, all of these questions really were based on the observation and the visits that we had with our colleagues and artisans in Mexico.
I just want to add that, a lot of the literature in Spanish. And so for me, that's been generally inaccessible. I don't speak Spanish. I don't read Spanish. But even when you're making an effort to do that type of research, the written documentation does not really translate well what's actually the artisans are doing. So to go and observe was so key in how it changed my perspective of like, what is lacquerware as opposed to what is a painted wooden object. There's a real difference, and you don't appreciate it until you go and see. So I think for a lot of these materials where there's not great written descriptions, or descriptions maybe someone like me can understand, going to observe is really, really key.
Thank you. So we have time for our last question, and I'll invite you all to speak more with our speakers after the break.
Okay, thank you. That was such a great, is it gone? Okay. I had a question actually based on the literature that you found. And I noted that, or you had mentioned something about the smell of the chia oil when it was burning. And so I was wondering if you noticed that during the workshops or even during, did that guide any of the analytical techniques that you decided to take?
I think, that the Alzate quote, that's what you're talking about, which was extremely fascinating as a conservator to read. A lot of the references that I presented have been, we didn't find them. Those have been republished before by my colleagues, and just make a note there. But in terms of burning and smelling, we would never do that to a painting. And it's very likely that Alzate was somewhat biased in his approach. He wanted to prove certain Mexicanity of artworks and also establishing a certain, sense of importance to chia oil as a Mexican lacquerware. As a Mexican material to paint and actually better than linseed oil, which was at the time imported. Also, he's writing later in the 18th century with a lot of these criollo feelings are starting to boil, and their ideas of independence there and all of these other things. So it's very interesting. I wonder if he was smelling the toasty smell of chia seeds. Some of the artisans that gave us some of the oils they prepare, they will toast the seeds until they would pop like popcorn, and the smell was quite strong and quite burnt, almost like burnt coffee. And I'm wondering if that's what he's alluding to when he's smelling these burnt pieces of paint.
Did you analyze the esters in that vapor coming up?
We didn't burn anything.
I think there hasn't been VOC.
On the seeds.
We haven't done any VOC analysis, and I know there's a lot of interest in analyzing the smell of things. But to be honest, since there's so many different recipes, especially for lacquerware, I'm not sure that'll be like a fingerprint kind of identification. There's a lot of things that will smell. And also, we never really explored all the variations of linseed oil recipes. There's a lot of work still being done on that. So I would be cautionary to identify something by smell alone.
Okay. So I would like to thank our speakers again. So again, I'll invite you all to ask some questions maybe during the break. Make sure they have coffee in hand first. And I'm going to introduce, yes. And also to listen to the Immaterial podcast on chia. That's on the Met website. Before we go to break, we have one more set of remarks from Al Carver-Cubic, who is a senior program officer in the Division of Preservation and Access at the National Endowment for the Humanities. Before joining the NEH, he was a researcher at the Image Permanence Institute at the Rochester Institute of Technology, where he specialized in researching printed materials and environmentally sustainable preservation solutions. So I'll invite Al up, and I'll ask the speakers to return to their seats.
Thank you so much. I won't be long. I don't want to be the guy that stands between you and lunch. So I just want to say a special thanks to the Metropolitan Museum of Art for organizing this symposium, and a special thanks to Julie for inviting me. The work presented this morning was supported by a Research and Development Award, which is administered out of NEH's Division of Preservation and Access. Research and development supports projects that address major challenges in preserving or providing access to humanities collections and resources. These challenges include the need to find better ways to preserve materials of critical importance to the nation's cultural heritage, and to develop advanced modes of organizing, searching, discovering, and using such materials. NEH's commitment to this essential work predates the R&D program, which was launched in 2006 and extends as far back as the 1970s and 80s. It is telling to see how far we have come from those early days to where we are today with the work showcased here, which in many ways represents an exciting future direction for the field. In the early years of NEH, R&D supported the development of critical instruments, equipment, and methodologies used for preservation work. We supported the development of the Preservation Environment Monitor Data Logger, which is still used to conduct collections assessments and monitor storage environments, including for small heritage institutions, and the creation of the Emergency Response and Salvage Wheel, which has been translated into numerous languages and is globally deployed. As essential as these projects have been to the preservation field, the last decade or so has demonstrated how far we still must go to address the needs of all communities. As research and development has continued to grow and adapt, it has supported projects to empower Indigenous and Native communities in the stewardship of their collections, improve accessibility for persons with disabilities, conduct studies of minority-serving institutions, and enhance access for collections representing the LGBTQ+ community. The message from the field was clear that Western-centered approaches alone would not suffice in preserving our diverse heritage. An NEH-supported national study on the conservation field conducted by the Foundation for Advancement in Conservation, called Held in Trust, confirmed what we have been seeing in our consultations, field analysis, and project review. In particular, the study on science and materials calls for interdisciplinary collaborations between science and art that go beyond European and North American makers to encompass all cultures and geographies. The first focus area of the study centers on innovation, stating the goal of innovation in heritage science is to positively address pressing changes that are unique to preserving and understanding cultural heritage, especially in achieving ever more precise identification of materials, assessment of condition, development of sustainable treatments, and, in collaboration with others, a more nuanced understanding of the creation and history of objects and sites. The findings from Held in Trust brings us back to your groundbreaking work. Your collaborative interdisciplinary approach to this research serves as a model for cultural heritage material investigation and sets a new standard for material analysis. The ability to identify materials used to create these objects is central to conservation and preservation practices, as well as expanding our understanding of the peoples and cultures who made and used them. Your project has deepened our knowledge of previously understudied and decontextualized objects from the Viceroyalty of New Spain, as well as from Mexico, from pre-colonization to the present. Bringing together novel scientific approaches with historical research and traditional knowledge places your research on the forefront of conservation science and museum practices. Of note, your project not only looks to draw on traditional knowledge, but I can see through your collaborations how your work seeks to elevate and meaningfully support communities in Mexico in their efforts to revitalize traditional lacquer techniques. While the research presented here is specific to the use of chia oils, as clearly demonstrated, the methods have widespread application and ought to serve as a model for future heritage science endeavors. Heritage science is at its best when the research is truly interdisciplinary, a collaboration among humanists and scientists of all varieties. What is new in heritage science is the integration of source communities. The challenge is not only to acknowledge the needs of source communities, but to involve them as active participants, even partners, in the pursuit of knowledge. As we see in today's symposium, this is not only ethical, but is essential to experimental work. The METS project has motivated us in Preservation and Access to continue to explore how our programs may support applied research that blends Western and Indigenous epistemologies, forges partnerships among diverse organizations, and unlocks access to understudied humanities collections located in the United States and around the world. Please know that we are always open to continuing the conversation about these shared interests. And thank you so much again for the invitation. And I've learned a ton this morning, as I'm sure you all have, and I'm really looking forward to this afternoon's presentations.
Okay, everyone. So the next session, session 2, will start promptly at 2 p.m. So please return to this room or the overflow room at 2 p.m. Thank you.
Okay, the lights are dimming, so I think that's my cue to get started. And I'd like to start with an announcement. For those of you who are in need of simultaneous translation into either English or Spanish this afternoon, headsets are available, so please make use of them if you need them. They're in the back in the corner here. Okay. My name is Rhonda Castle. I'm the curator of Latin American art here at the Met. I often joke that my role as a member of this research team is to be the curatorial mascot. I'm really thrilled to be here this afternoon and to have the opportunity to introduce the speakers in the second session of the symposium, which is titled From Cultigen to Art Material. The first speaker this afternoon is Dr. David Lentz. He's a professor in the Department of Biological Sciences at the University of Cincinnati. He's the author of nearly 130 peer-reviewed articles and 3 books, including Imperfect Balance, Landscape and Transformations in the Pre-Columbian Americas, Seeds of Central America and Southern Mexico, and Tikal, Paleoecology of an Ancient American City. This afternoon, Dr. Lentz will speak about chia as a Mesoamerican cultigen, its dietary, medicinal, and ceremonial significance. Please join me in welcoming him.
Thank you all for being here. It's a pleasure for me. I would like to thank the organizers, Rhonda, Julie, Jose, Fran, and many, many others. It was very kind of you to invite me to participate in this project and for putting together such a knowledgeable contingent of speakers focusing on a timely and intriguing subject. Our topic today is chia, an herbaceous annual. Let's move along here. An herbaceous annual in the Lamiaceae or mint family. The Latin name for chia is Salvia hispanica. On the left of this slide, you see the colorful flowers on the spike of a chia in fluorescence. Each spike will produce dozens of seeds, and a field of chia, as you see on your right, will produce millions. Chia is a well-known crop in Mexico and Guatemala, and its native range is shown on this image. The name chia comes from the Aztec word, or Nahuatl, meaning oily seed. And the Aztecs greatly prized this plant and used it for many different purposes. As you can see here, there are 9 other species in the genus Salvia, however, that are also called chia, and they are illustrated before you. Note that there are 2 other unrelated species, the Amaranthus hypochondriacus and the Hyptis suaveolens, but these also have oily seeds and are often referred to as chia. In particular, the amaranthus is noteworthy because they have the blood red flowers, as you can see. And this is a feature that gave rise to its association with Aztec bloodletting rituals. And we'll talk a little bit about that in a moment. So, why was Salvia hispanica widely favored among all of the other plants called chia? The likely answer is that it is much more productive in terms of oil output than the other chias. As you can see in this table, the amount of oil produced by Salvia hispanica far exceeds the other chias, especially the Hyptis and Amaranthus species. And I want to point out some amazing things about this too, is not only does Salvia produce a lot of oil, but it also produces oil that's of very high quality with omega-3 fatty acids in it. Linoleic is especially good, and these are all polyunsaturated. So these are very high quality oils. And as we read about this more and more, I think you're going to see that it's amazing that we don't use this plant more than we do. But I think the health food craze is helping to change all of that, fortunately. But you can see like the Hyptis suaveolens and the Amaranthus, they're nowhere near as productive in terms of oil and in terms of the quality of the oil. Now this table compares well, we've already seen a table that compares the amount of fats, proteins, and carbohydrates of all the different chias. This image shows the small size of chia seeds as compared to a dime on the right. And this was a former governor of New York there. He's not in the picture yet. Okay. All right, there is our former governor and former president. And these are very small seeds, but their productivity per acre is comparable to other crops that we're very used to talking about. Now here we see illustrated one of the unusual properties of chia. Associated with the seed coat, there are numerous cells that exude a polysaccharide that creates a mucilaginous cocoon around the seed. Now, this is an adaptation to the arid environments where chia commonly lives. The cocoon will keep the seeds moist and help them germinate even if rainfall becomes scarce. But, you know, in looking at this, people generally, their typical reaction is, oh, yuck. I mean, that looks awful. But, you know, if you mix this with some berries and a little almond milk, maybe throw in a little ice cream, it makes a terrific smoothie. So I highly recommend it. And it's extremely nutritious, as we have seen. Now, the plant... Let's go ahead. The plant was domesticated somewhere in northern Mexico around and possibly before 3,000 years ago. The earliest archaeological discovery of chia came from the late archaic site of Cerro Huanacaña in the Mexican state of Chihuahua. More recent discoveries of archaeological chia were unearthed at the Tlatelolco site in central Mexico in deposits that date to late formative times, that's about 400 BC. Thereafter, the identification of chia in central Mexican archaeological contexts becomes much more frequent, indicating that the use of the plant expanded rapidly during the Classic period, which went from AD 200 to AD 900, and the Post-Classic period, which went from AD 900 to 1520. Now, this is the Temple of the Sun at Teotihuacan. This is, or was, the largest structure in the Americas until the 18th century. It was built by the Teotihuanacos, who were the dominant cultural force in Mesoamerica during the Classic period from AD 100 to 600. Chia was found in many archaeological contexts at this site and in adjacent sites. So this was a very popular thing during the Classic period. Now, following the collapse of the city of Teotihuacan, the powerful Aztec Empire, which extended from the Gulf of Mexico to the Pacific Ocean, emerged around AD 1300. Chia was an important crop for the Aztecs, who used it for many different purposes. Okay, a few years ago, building contractors were digging in Mexico City. They came across the remains of an Aztec temple, the Temple Mayor. And you know that Mexico City is built over the ruins of Tenochtitlan. So underneath the city of Mexico, there are the ruins of the Aztec people. And oftentimes when they try to build something, they find ruins underneath. But since the discovery of the Templo Mayor, the archaeological excavations have ensued, and the scholars have gained further evidence that Chia was used extensively by the Aztecs. Now, here's a discussion of how the Aztecs used chia for food. One way, the seeds were ground to make a flour, as illustrated here, to make a chia pinole for tortillas, tamales, etc. The oil from seeds was used for cooking, and a refreshing beverage is made directly from seeds. It's called agua de chia. And if you have the seeds, even with their mucilaginous coating, it has sort of a lemony flavor to it. So it's much better than it looks. And also the Maya, or the Aztec warriors, carried bags of chia seeds with them on the trail, and they would mix it with agave syrup. And because of the high fat content and the high protein content, this is a very effective trail mix for them. Now, tribute records show that chia, along with maize, beans, and amaranth, were the principal foodstuffs of the Aztecs. Now, one of the records that we have in abundance from the Aztec period are the tribute records. And these records are a recording of all of the tribute that the surrounding villages had to bring into the Tenochtitlan. And the way it works here is that over here we have, these are the names of all the villages, and then over here and over here, this is the stuff they had to bring in to the Aztecs every year. And over here we see these wooden boxes, and people were required to bring in crates of food, and this was the chia and maize, and this is beans and amaranth. I don't know why they have them together, but the total income of chia for the Aztecs in 1 year would be 4,000 tons. 4,000 tons with a T of chia. So this is really big food. There would be 7,000 tons of maize that would be brought in. So this was really phenomenal, very important food stuff for the Aztecs of Tenochtitlan. And you kind of wonder, where did all this stuff come from? And one of the answers is a lot of this was grown in what they call chinampas. Has anybody here been to Mexico City? I'm sure a lot of people. Yeah. Has anybody been to Xochimilco? Yeah. Okay, did you take one of the boat rides there? Yeah. Up and down the canals. They're fun, aren't they? But Anyway, so this is what the Chinampas look like. This is an image of the Chinampas from 1900 that in the archives of the Lloyd Library, which we have in Cincinnati, one of the founders, Curtis Lloyd, went down to Mexico and he took all these photographs in 1900. So this was a something from that record that gives you an idea what things looked like in 1900. But these were called chinampas. Some people call them raised fields. Some people call them floating gardens, which they're not floating. But the way they made them is this is a kind of a swampy area, and they would dig a canal, and then they would pile the soil up in the center, and they sort of would build fields. And this is actually a very highly productive means of agriculture because there was always a source of water in the ground. The ground was always moist, and you would always get a crop. And chia, it turns out, was one of the major crops of the Chinampas during Aztec times. Also, here's another picture of the Chinampas. The Chinampas were good not only for raising chia, but they were good for moving goods around. They could use them as canals, and also they would use them for fishing. So there would be fish in the canal. So it was really a very highly productive system of agriculture. Now, also, chia was employed in an impressive array of medicinal applications. Just a few of them. Females consumed ground chia seeds mixed with roasted maize, and they would include a possum's tail. And this concoction would induce labor, and this is what is pictured here in the Florentine Codex. By the way, these codices, each codex was a book that was authored, written, painted by Aztec authors. So this gives us these books give us a real insight to what life was like among the Aztecs. Also, they used the oil as a treatment for burns, and this seems like a very logical way, a very logical thing to do, that must have been a very effective treatment to have put that oil on a burn. Other things, the seeds were used to remove obstructions from the eye, and also it was a good treatment for eye infections. Now, as we've heard this morning, the seeds contain a superb drying oil, and it's served as a solvent for paints or as coatings for ceramic vessels and gourds, as seen in this image here. Once again, that's the Florentine Codex, and there's a person putting a coating on a ceramic vessel, and this is just something from my own personal collection from one of my many trips to Mexico. And this is Crescentia cuyete, or calabash, that was had a coating on it of chia oil. You can see it has a nice glossy coating, and chia is very well noted for that. Okay, in terms of ceremonial uses of chia, the Aztecs crushed the seeds and prepared it as an ingredient in zoayi, a kind of confection mixed with amaranth and maguey syrup. And you can see it here in the middle. And this, during certain ceremonial times of the year, they would fashion this so that it looked like their god Huitzilopochtli, who was their god, the sun god of war and of human sacrifice. Okay, we're down to 5 minutes. Holy cow. Anyway, so you can imagine the Spanish friars were not too happy about this association. They tried to convert the Aztecs to Christianity, and they tried to eliminate the connections with their traditional religion. And unfortunately, because amaranth and chia was associated with this, This, according to some authors, may be why the growth of chia in the viceregal times went way down. It was not the prominent crop that it was during Aztec times. And there are other factors as well. One of them is the introduction of Eurasian crops. And also there was a 16th century population crash that caused a sharp decline in the population and in the production of these two valuable Mesoamerican crops during the viceregal times. Because of their undeniable value, however, both amaranth and chia have made a significant comeback in recent decades. Now, we have talked at length about how the Aztecs used chia, but how did the Maya use chia? Actually, there's very little evidence that the ancient Maya did use chia, yet they must have known about the cultigen because they traded extensively with Teotihuacan. And it's possible that we just don't find the evidence because the seeds don't preserve well in the wet and dry tropics. Current research at the Calakmul site may change our ability to detect chia, however. Now, Calakmul, this is the location of Calakmul, smack in the middle of the Yucatan Peninsula. It was one of the leading centers of the ancient Maya world. Now, this is one of the largest temples at Calakmul, and it illustrates the forest surrounding the area. This is in the middle of the Calakmul Biosphere Reserve, and it's a wonderful place to work, botanically speaking. Okay, now this is a LiDAR image of the ceremonial site core, showing temples, palaces, reservoirs, ball courts, and marketplaces. South of the site core, we see dense areas of walled fields and agricultural terraces. Even further south of the terraces, we find a large drained field complex reminiscent of the Xochimilco chinampas. It is possible that the ancient Maya used this field or the terraces to grow chia. Our team aims, by using newly developed environmental DNA techniques, to learn exactly what crops were being grown in the terraces and drained fields of Calakmul when we return to the site next spring. In summary, I didn't show you this one. This is the drained field complex. And if you look very carefully, you can see all of the canals that go throughout this drained field. And there are just a few houses around it. So the productivity of these drained fields must have far exceeded what was needed for the local people to sustain their families. Therefore, this was a place where there would be a large productivity, and it would have helped to explain how the people of Calakmul were fed. Okay, in summary, there were many species of plants called chia in Mexico, but Salvia hispanica is the most productive and most nutritious, and hence the most widely used. The earliest evidence of domesticated chia was found in archaeological contexts from around 3000 BP, but domestication probably occurred much earlier. The ancient center of Teotihuacan and surrounding settlements show that chia evidence dates back to at least 400 BC there and expanded dramatically thereafter. The uses of chia by the Aztecs has been well documented historically and ethnographically. The Aztecs used chia for food, medicine, paints, coatings, and ceremonial purposes. The use of chia declined significantly during viceregal times, probably because of the introduction of new crops, the population crash of the 16th century, and the antipathy of Christian clerics towards plants associated with indigenous rituals. And finally, future efforts at Calakmul likely will yield new insights into Maya agricultural practices, possibly involving chia production. Thank you very much.
Thank you very much, David. The next speaker this afternoon is Dr. Gerardo Gutierrez, Professor of Anthropology at the University of Colorado Boulder. Please join me in welcoming him. Dr. Gutierrez's research focuses on the archaeology, ethnohistory, and technology of pre-Hispanic and early colonial peoples of Spanish America, with special attention to the with particular attention to the archaeology and codices of the Mixtec, Tlapanec, and Nahua kingdoms of Eastern Guerrero and the Huastec region, and political systems of the Gulf of Mexico. In his presentation this afternoon, Dr. Gutierrez will share his research on early lacquer technologies and evaluate evidence derived from the study of a decorated gourd fragment found in Honduras in the El Gigante rock shelter. Please welcome Dr. Gutierrez.
Thank you very much, and thank you to the organizers for the kind invitation. So this is forward, and this is backwards, and there is our laser. All right, very good. Lacquer indigenous technologies in the New World have used different organic polymers to apply and fix color coatings. For Mexico and Guatemala, chia oil, Salvia hispanica, or the fat of the aje or nig, as it's called in the Maya area, and a scale insect with the scientific name something like Yabelia ashin, is well reported. While in Ecuador and Peru, the mopa mopa resin was used. Nevertheless, there is little information of what type of lacquer technology was used in the intermediate regions from Honduras to Panama. A surviving fragment of a decorated gourd found in a Gigante rock shelter in Honduras sheds some light into this issue and offers insights for understanding geographic and chronological changes in Native American lacquer technologies and gourd decoration, of course. This is a research collaboration with Dr. Ken Hurst of Pennsylvania State University, who provided the sample. What happened here? Pretty much I'm trying to put here together all the different areas with the different polymers. And also I'm going to analyze the minerals just to give context to what is going on here in Honduras, right? The technology here resembles the technology here, or it resembles technology from farther north. Okay. Lacquerwork seals pigments and dyes with organic polymers. We examine similarities and differences of various lacquer production centers to help us understand a decorated gourd from an early context in Honduras. Reviewing key processes of this Mesoamerican lacquer craft can aid in better understanding how knowledge was acquired and passed through communities of practice. We approach this analysis by distinguishing the different regions of production based on the type of sealing polymer used, primarily if the communities of practice use a drying oil or the fat of the aje scale insect. Lipids polymerize differently and can or cannot bind specific mineral pigments and dyes. Therefore, they can impact the change of operation, aesthetics, and durability of the lacquers, and the overall cost of production. And here we can see some experiments that are done with aje, of course, with the different oils that there are more oils that were used, but of course the chia and then the European linseed oil are the ones that are more accessible, right? It's like other local oils are difficult to find. And you can see that the instrument, sometimes they don't tell us the difference between one thing or another, but the molecules reorganize during the polymerization and actually create different textures. And in the case of the aje, you can see the molecular size of the fat that the insect provides. And that's the kind of 1 kilo aje that was shown in Guatemala in a recent trip. And this is the aje from Chiapa de Corzo, of course. So experimentation is key, is a key component for, okay, we look at certain materials and their transformations during different steps of production, monitoring them by measuring their molecular interaction with different frequencies of energy. For this, we have collected in the field many raw materials used from various locales to compare them with laboratory references. Experimentation is key. It's a key component of this research, especially to better explain key processes, but mostly to rediscover why communities of practice selected the properties of each material and chose some over others. Then why they get discarded by modern practices only to return to these traditional materials over and over again. I mean, it's like in the last 50 years, new materials have arrived. People can use them for some years, and then they decided that they are not good enough. They go back to the traditional materials. Additionally, we study the diversity of materials used in each production center, under the premise that the selection of minerals is deeply imbricated with local geologies, but also with the polymer used in each region. For this, I have visited and examined all the key production centers of Mesoamerica to collect samples of raw materials and objects produced by the key masters of each center. This methodology helps to detect outdated characterizations perpetuated in bibliographies, given the myriad of names that change from region to region, but even inside different workshops of a production center. For instance, the repeated belief that dolomite is or was the key mineral in the production of lacquer, which is not, by the way. So what we have is pretty much calcite and gypsum and also volcanic ash. Okay. The lacquer centers of Olinalá and Temalacatzingo provide almost ideal conditions for the study of lacquer, simply by the sheer number of workshops and masters present in both towns, to the extent that a large percentage of the population of those towns have worked at one point of their lives in one of the many segments of production. The Cochle, let's see the materials they have. The Cochle is an iron-rich kaolinite, is a mineral that absorbs the chia oil to produce the creamy paste used to start the process. Sorry, I got lost. The Cochle is the iron-rich kaolinite, is a mineral that absorbs the chia oil to produce the creamy paste used to start the process of lacquer production in Guerrero. In Guerrero, the aje is not used. The craftsmen claim that the aje does not glue the local materials. Before entering into debates, it is important to note that the lacquers from Olinalá and Temalacatzingo come from different lacquer technologies than those found in other centers. It is not better or more beautiful than those produced in Michoacán, Chiapas, and Guatemala. It is just different and perhaps easier to produce, reducing costs in time, labor, and materials due to the economies of scale, division of labor, and the specialization reached by reaching these two centers of Guerrero. In this lacquer technology, the Tecochle receives the tlalpilole. Tlalpilole is just a mixture of a hemihydrated gypsum, also known as basanite, toctetl, that's the ground calcite, and tessicaltetl. Toctetl, that's the gypsum, basanite, and the ground calcite, that's the tessicaltetl, and other colors that will be used. Further decoration is easily applied on the soft layers of kaolin and gypsum before the chia oil polymerizes and hardens. And as they were explaining, the thing is that to compress really the layer of color into the base that is formed by this creamy chia oil with this kaolinite, rich in iron. If we were talking about Mesoamerican architecture, the cycle of limestone would be of utmost importance. That's how limestone can be burned and become dusty, and then we add water and hardens again in the shape that we want, right? But in the case of the Guerrero lacquers, the cycle of the gypsum from rock to basanite, anhydrite, and back to a fully rehydrated rock is actually more important and key. The drying oil of chia and the water trapped in the mucilage produce both a rehydration of the hemihydrated basanite back to rock, as well as a 3D polymer film that protects the lacquer. So it's like both processes are key. And it would have been impossible to use if we were use only oil without the mucilage. The mucilage is needed because it carries enough water to do that rehydration of the gypsum. Other relevant technologies are those found in Chiapa de Corzo. Other relevant lacquer technologies are those found in Chiapa de Corzo, Uruapan, and Pascuaro, which are based on volcanic tuffs weathered into montmorillonites, rich in iron oxide and calcite, fixed with aje fat or a combination of aje and linseed oil called sisa. Aje dries very fast, and together with the harder volcanic minerals, it is less forgiving for corrections if mistakes are made. For reasons related to regional macroeconomic processes, it is more difficult for Michoacán and Chiapas to maintain many masters working on the craft. Nevertheless, I was happy to find that the indefatigable Navarro Sanfale family is making inroads in experimentation with multiple minerals, including kaolinites. This is one of Tomas' experiments, and he was very kind to provide it to me. And this is just with the use of optical absorption. And we can see how the main components is, of course, the chia oil that is there. The sisa of chia oil and aje is there, and the kaolin, and then, of course, the rich kaolin in red with iron, the iron, and of course, then the final spectra of this example. The craftsmen of Rabinal, Guatemala have surpassed this issue of dealing between minerals and oils and the aje by specializing in gourds decorated only with aje and black soot, carving motifs directly on the gourd. And if you want colorful lacquer in Rabinal, you just buy the painted one with automotive lacquer, right? Because you can buy it for $3 the piece, right? This takes us farther north to the borders between Nayarit and Jalisco in the Huichol region that we have, where naturally occurring green aluminum silicate clays rich in calcium and iron, possibly produced by hydrothermal processes, plus the red kaolin-rich clays, clays rich in iron oxide, are used in the decoration of the Huichol gourds. Gourd production in the Huichol region continues to be based on a complex religious ideology around the long duration of hunting for peyote. Huichol lacquer masters do not add gypsum or extra calcite to their chia to their lacquers and they use only shea oil and the materials that I have just told you. And the decoration is created based on mythical events or visions that they had during their peyote hunting, and then decorated with glass beads glue with beeswax. Perhaps the Huicholes are the only producers of lacquer who are not worried about achieving lacas that can last for 20 years, for 100 years, or for 400 years, right? For them, the lacas have a cycle and a purpose. And once it is achieved, the lacas need to degrade and reintegrate to the world. After this long detour, we can see the relevance for studying a tiny sample of a decorated gourd from Honduras. This was found in El Gigante cave that is around 100 kilometers from Tegucigalpa and 50 kilometers from the border of El Salvador. It is a small fragment of a curcubita ring of 7 by 4 millimeters, exhibiting a beautiful bright red color, although with serious damages. It was found in a cave with an occupation beginning at 10,000 BC and with some dates closer to 1500 AD. The occupation of El Gigante was marked by long hiatuses with abandonment at 200 AD. So that gives us the opportunity to guess, because we are going to do the radiocarbon dating after we finish all the testing, but that gives us the opportunity to guess that this decoration happened between the early Estanzuela phase or the late Estanzuela phase. And here we see other archaeological gourds that have been decorated gourds that have been located in Mesoamerica, and the date, which actually corresponds well with the proposal of David of 3000 BC domestication of the chia seed. So therefore, the decorated gourd under study likely dates either between these 2000 to 850 BC or 400 BC and 200 AD. This decorated gourd may be contemporaneous with other early archaeological gourds, especially that of Tlapacoya, right? It's like, but we will see until we date it. If not, I'm happy if it's Chupicuaro, right? Too. Interestingly, the gourd of Tlapacoya is lost in the collections of the Museum of Anthropology. So we need someone to go and look for it. The elemental analysis shows iron-rich red color, and its UV confirms yet another red ochre mix with a kaolinite. The decoration of hematite and kaolin is layered and well embedded into the cellular structure, which means that most of the damage is due to the slow disintegration of the gourd ring itself and not to the failure of the pigment. The kaolinite iron oxide pigment was well corroborated for the FTIR study, but we were also interested at this point in knowing if a lipid was used in its manufacture. And unfortunately, we only had like very weak shoulders in the area where we were supposed to find presence of oil. Nevertheless, we run contact Raman in the, that's by the way, just the explanation of the kaolinite and hematite in the sample. So we run a contact Raman, and it gives us a spectrum that at least in a timid way has peaks in the area where we're supposed to find oils, right? And it doesn't look like or or doesn't have evidence of the peaks where we are finding or tend to find the lacquer, the AGFAT. For these, we needed to continue corroborating the existence or not of a possible polymer, right? So for this, we prepared samples of pure dried aje and chia oil to use the near spectroscopy. And we actually found that at least in this spectrum segment, actually the aje and the chia, they do have slightly different values. Okay. So once we did that, we went to actually objects that we have acquired among different workshops in Mexico and tried to see if we could detect the same. And indeed, for this mask from Tamalacatzingo, we detected exactly the existence of the lipid. Then we tried another object. This was done by the Navarro family in Uruapan, and indeed we can find that it presents the peaks of absorption for the lipid, in addition to peaks of absorption for a clay. That was interesting. So, but when we did that for the sample of Honduras, we did not find this peak, this absorption here, and here we're just less than optimal, which just could be that the lipid is aged, or we have to face that perhaps the lipid is not there, right? A preliminary analysis, and my colleague told me that he was going to send me the results, and yesterday, and it's today, and I checked my email, and they have not arrived. But a preliminary analysis with lips is giving us the presence of, in the surface of red ochre, is giving us the presence of carbon, oxygen, and hydrogen. So I still have some hope, right? So otherwise, perhaps I can give you the fragment to you guys, and you can like solve the mystery. All right. Conclusions. The decorated surface of the GERD from El Gigante Honduras exhibits a layering of red ochre, hematite mixed with kaolinite. There is no evidence for the use of calcium sulfate or calcium carbonate. Like in the case of Guerrero, there is no presence of degraded volcanic ash or calcium carbonate like the Tizate of Chiapa de Corzo or the Charanda of Uruapan. Technologically, the El Gigante lacquer is closest to the lacquer produced by the historical Huicholes. If further testing confirms the use of chia oil in the lacquer or the decorated gigantic, okay. If further testing confirms the use of chia oil in the lacquer of the decorated El Gigante gourd, it offers further insight into the development and geographic distribution of the New World lacquer technologies, in particular, a north-south regional intercalation between the use of A, aje, with degraded volcanic ashes versus B, chia oil mixed with gypsum and kaolinite. This complex geographic intercalation would indicate a reassessment is required for understanding the networking of communities of practice in Mesoamerican lacquer production in the pre-Columbian and colonial period. If no lipid is confirmed in the El Gigante sample, then it would suggest that this decorated gourd surface shares a technological tradition more related to pigments seen in early rock art, where kaolin clay provides adhesiveness to the painted surface. Thank you very much.
Thank you very much, Gerardo. The final speaker in this afternoon session, the first of two afternoon sessions, is Pablo Amador Marrero, who is a researcher at the Instituto de Investigaciones Estéticas at the National Autonomous University in Mexico. He is an accomplished art historian as well as a conservator. Dr. Amador's research, undertaken in collaboration with his colleague Elsa Arroyo, also from the UNAM, centers on the materiality of artworks, with a particular focus on polychrome sculpture. His extensive study of the export and reception of lightweight sculpture made of materials like cornstalk paste is especially noteworthy. He is the curator of numerous exhibitions, most recently having collaborated on the Prado exhibition Tornaviaje, Arte Iberoamericano en España in 2022-2023. This afternoon, his presentation contextualizes the documented use of chia oil in connection with the painting of consecration crosses and other artworks in Puebla Cathedral in 1649. Welcome, Pablo Amador.
Thank you. Thank you, Rona. Siento no utilizar el inglés, pero mejor por ustedes. Quiero dar las gracias, por supuesto, a la institución, a Julia, a José Luis, por la invitación. Y lo que sí me gustaría empezar es comentando que yo vengo de una institución, el Instituto de Investigaciones Estéticas, que nos dedicamos al arte. Todas nuestras preguntas van vinculadas a la historia del arte. Nosotros hacemos cuestionamientos desde la materialidad sobre el arte y volvemos al arte para resolver esos o intentar resolver esos cuestionamientos. Y hoy la verdad es que cuando se me invitó, yo estoy ahora preparando la exposición de Luisa Roldán y como que la chía entraba en mi vida y salía. Y entonces cuando hace algunos meses se me invitó, volver a la chía me llevó también a volver a pensar problemas que teníamos sobre la historia del arte. Entonces, el día de hoy, lo que mi conferencia o mi ponencia se va a centrar en 3 puntos. En otros orgánicos, nosotros tenemos un problema en la universidad, en nuestra institución, nuestro talón de Aquiles, nuestro punto débil son los orgánicos, no tenemos el cromatógrafo, no hemos sabido hacer buenas alianzas con otros compañeros para desarrollar el estudio de todos estos materiales orgánicos, pero lo intentamos solucionar con ingenio, como buenos mexicanos y más como buen canario que soy yo. La otra parte que les voy a plantear es cuando nos hemos encontrado la chía, qué ha pasado con esa chía que hemos encontrado. Y por último, lo que sí nos gustaría es como plantear ir detrás de la chía de nuevo. Y volver a la chía nos va a hacer volver a nuestros datos, porque como decían, ese documento de origen apareció a partir de un archivo y los archivos son fuentes fundamentales para la historia del arte, al igual que los propios objetos artísticos. Siento no traerles tanto gráfico y tanto estudio molecular, pero es que no sé de ello. Yo me dedico a la historia del arte. ¿Qué orgánicos tenemos? ¿Qué orgánicos nos hemos encontrado? ¿Con qué orgánicos nos hemos planteado hacer preguntas? Pues empezamos con las piezas iniciales. Este es el Cristo de Bornos, es un Cristo de caña de maíz, un Cristo hueco de caña de maíz, con una historia vinculada a Michoacán, de donde vienen las bateas, pero una historia con caña de maíz y con orquídeas, pero que no tiene orquídeas. Y nos ha interesado ese aglutinante, porque la orquídea, al igual que la forma de construir las imágenes macizas y no huecas, nos está dando desde la historia del arte una ruptura con la tradición del arte americano. Y concretamente en plantear que los Cristos de caña no son solo de Michoacán, sino también son del centro de México, son de la Ciudad de México, y donde los Cristos de la Ciudad de México están mirando a Europa para su construcción. Son imágenes como las que estaban haciendo en Bélgica, en Flandes, como las que estaban haciendo en Italia o como las que se estaban haciendo en España, pero añadiéndole caña de maíz, un material propio. Mientras que estas, que son las de Michoacán, sí tienen orquídeas, y ese es el fragmento que les presento en la parte baja, tienen orquídeas que hablan de los lugares, que hablan de geografías, que es algo que a mí me ha interesado siempre, la distribución geográfica del arte. Y es que, pero no solamente nos ha dicho eso, sino que la restauración del Cristo en el Instituto Andaluz del Patrimonio Histórico nos llevó a encontrar un códice como parte del paño de pureza, y en ese códice lo que vamos a encontrar es que no está hecho con el aglutinante, la cola de origen animal, o sea, el pegamento habitual, la cola de carnero, la cola de retazo, sino que estamos encontrando un almidón para pegar. Ya no solamente no es orquídea, sino que tampoco es la cola habitual de conejo. Y eso nos está hablando que aquí la están usando para un caso concreto, que es pegar fragmentos de códice de reutilización para hacer un paño, el paño que viste a Cristo. Y este es el pequeño... Es un códice de reutilización. Y este es el fragmento que está hablando de ese aglutinante diferente, porque los aglutinantes también nos están diciendo sobre fórmulas aprendidas, y en este caso muy probablemente con maestros también europeos. Pero esto, que fue hace más de 15 años, se conecta con una intervención realizada en la Escuela Nacional de Conservación y Restauración del INAH, en la Ciudad de México, en la que colaboré como profesor, y donde otro Cristo de caña lo que nos enseñó es que no solamente estaban utilizando esa cola, ese aglutinante, para pegar los papeles, sino también para pegar fragmentos de caña. Y eso lo que nos están diciendo es que tienen una práctica en donde esta cola pega más rápido y no deforma la caña, los fragmentos de caña para hacer las imágenes. Pero nuestras aproximaciones, y esto se lo debo a mi compañera, también se deben desde otros puntos de vista a los orgánicos. Y nos interesa especialmente este proyecto, es un proyecto sobre el azul. Mi compañera del desarrollo está muy interesada en el color azul, igual es tendencia, ténganlo en cuenta para ahora comprar en las Navidades. Pero es importante cómo la abordamos nosotros. La abordamos desde la praxis, desde el conocimiento del arte, pero también desde la Technical Art History. Hacemos toda la praxis de construir o de construir los procesos técnicos de manufactura de los objetos. ¿Para qué? Para entenderlos mejor y contextualizarlos. Y en este caso es con el azul de Chiquilite. Sí vamos a... Es de los pocos que le voy a poner. Sí vamos a la instrumentación, pero vamos con otros tipos de preguntas. Nos interesa el proceso de desarrollo y nos interesa llevarlo a leer los objetos. Y esto nos lleva a la segunda, al segundo cuestionamiento de lo que hablamos hoy, el encuentro de chías. Y he puesto la batea porque a través de Cartella Fine Arts, que es con quien he colaborado en algunas ocasiones, han aparecido pues, la chía que ahora está siendo analizada por los propietarios de esta magnífica batea, ¿no? Que es esta institución y sus investigadores. Pero no me marcho de la misma galería y voy a un objeto que me tiene verdaderamente enloquecido, que es este gran baúl de la Villa Alta de Oaxaca. Nos vamos a Oaxaca en esta ocasión. Y con este baúl, que además es un baúl que rompe ciertas tradiciones porque en él se combinan diferentes iconografías que están hablando de talleres mucho más amplios, menos talleres en la Villa Alta, pero talleres más potentes, que unifica iconografías. De él nos interesa además, fíjense qué maravilloso baúl, viene de una colección francesa, y lo que nos interesa es el hallazgo, a partir de un pequeño deterioro, de lo que se denomina el sulaque. El sulaque es una pasta mítica que tiene infinidad de traducciones, tiene también literatura asociada, una literatura que habla de cómo se hacía, pero que la realidad científica es que está diciendo que así no se hacía. Que es lo que pasa muchas veces. Es como Pacheco. Pacheco te describió cómo había que pintar, pero a él se le olvidaba hacerlo. Todos aquellos que hemos trabajado Pacheco lo descubrimos, ¿no? Es muy difícil encontrar 6 capas de aparejo, o es muy difícil encontrar que te hagan 6 capas de yeso, porque es mucho más trabajo. Lo interesante de esta caja, de este baúl maravilloso y con un estado de conservación verdaderamente encomiable, Y cuando se le hacen preguntas, como la del Sulaque, es que el Sulaque nos está dando que es una serie tiene también partículas de agave, o sea, fragmentos de agave, tiene serrín, tiene cola, pero también tiene chía. ¿Y qué hace la chía en los baúles de Oaxaca? ¿Hasta dónde estamos llegando? Eso plantea algo, porque siguiendo la chía en los documentos, recientemente me encontré en otro documento que la chía se compraba, o sea, que el material con ese Sulaque, con chía, se podía comprar en las ferreterías del siglo XVIII de la Ciudad de México o de la ciudad de Puebla. Y hay una carta de pago que se dice que se vayan a comprar tantos materiales, tantos clavos, tantas cosas, y entre ellos Sulaque. O sea, se compraba también en las tiendas. Y el Sulaque con la chía y los materiales autóctonos y los materiales que también están mitificados muchas veces, que hemos creado una literatura que hay que revisar, como pasaba con los Cristos de caña, que eran verdaderamente las deidades prehispánicas, pero convertidas en españolas, lo cual no es verdad. También nos dicen otras cosas. Y nos vamos al tercer ejemplo, detrás de la chía. Tras las huellas de la chía, y los llevo a Puebla de Los Ángeles. Y a la Catedral Nueva de Puebla de Los Ángeles, una catedral que se inicia en el siglo XVI. Es una catedral que tarda mucho tiempo en hacerse, que se construye, que no gusta, que no hay dinero, que eso es también muy típico, que se quedan otra vez sin dinero, que también es otra vez típico, y que no hay dinero para seguir construyéndola, lo cual también es muy típico en cualquier construcción española o hispánica. Menos el Tren Maya, eso sí. Lo interesante de esta catedral es la llegada de, a quien tienen ustedes ahí, a Juan de Palafox y Mendoza, un personaje fundamental, hoy devoto, hoy es casi santo, que a su llegada se propuso acabar la catedral y en 10 años lo hizo. Él se la encontró en el primer tramo, no llegaban ni a los capiteles de las columnas. Y con el dinero, el obispado de Puebla es uno de los obispados, es el segundo obispado más importante de México, del amplio virreinato de la Nueva España, pero desde el punto de vista económico era igual de grande que el virreinato, el obispado de la Ciudad de México. O sea, el dinero que se recaudaba o que se donaba al obispado era enorme. Y eso favoreció que a la amada Puebla, como le llamaba él, a su amada ciudad y a su amada iglesia, dedicara gran parte de su riqueza, de lo que iba acumulando. Y Palafox fue también muy meticuloso a la hora de construir su catedral. La modificó, pero también se rodeó de un grupo fantástico de personajes que supieron entenderlos. Esto que les presento ahora es parte o resultado también de 2 años de mi vida dedicado a leer documentos en la Catedral de Puebla. Creo que de 1,5 dioptrías aproximadamente, que me multiplicó el COVID por 2, o sea, vamos juntos. Lo interesante es que cuando Palafox está acabando de construir la catedral, va a dividir cada capilla, la va a dividir y le va a hacer un encargo a uno de los miembros de su obispado. Cada uno va a tener que construir una de las capillas, pero va a traer con él en la embarcación y en su séquito, como buen príncipe de la iglesia, como buen rey de su iglesia, va a traer también a un artista, que es Ferrer. Ferrer es un religioso que aprende en Zaragoza y aprende siga a Carlos Borromeo, y él es el que va a ser el jefe de obra de la Catedral Poblana. Todo va a pasar por él. Y cuando la catedral está a punto de ser acabada, toca el momento de construir el altar mayor, construir el tabernáculo, pero también llega el momento de preparar la consagración del templo. Y todo esto hablan los documentos. Justamente en este documento que les presento es el inicio, simplemente es el inicio de cómo don Juan de Palafox y Mendoza pide que se empiecen a hacer los tabernáculos. Desde la primera piedra de Tecali, desde la primera piedra de alabastro desde el primer burro y desde el primer obrero que trabajó, está totalmente detallado en los volúmenes. ¿Cuántos carromatos viajaban diariamente desde las canteras a la catedral? ¿Quién los trajo? ¿Cuántos burros participaban? Todo está perfectamente detallado. En esos detalles, por supuesto, van a estar los artistas, pero van a aparecer más tarde. ¿Por qué? Porque hay que traer mucha piedra. Y piedra que se estropea, que hay que cambiar, que hay que utilizar nuevos materiales. Todo eso queda detallado. Y llega el momento de construir el gran retablo de los reyes. Y me interesa mucho este retablo porque este retablo está pintado por Ferrer. Las grandes pinturas están hechas por ese mismo personaje que trae Palafox. Y entre esa documentación, piensen los miles y miles de páginas que hay de construir una catedral. Vamos a encontrarnos verdaderas maravillas de documentos. Y entre esa documentación no solamente están los contratos de Ferrer, sino que Ferrer dirigía o controlaba ciertas obras. Y entre ellas algo tan sin poca sustancia como que, y aquí lo dice, está haciéndose cargo de mandar a comprar ciertas cosas, y aquí lo pueden ver, memoria de lo... espérense. Memoria de los encerados que se hizo de dar aceite y barniz. A ver, no escribo con tanta falta de ortografía, es como se escribía en el siglo XVII. Los cuales ellos están echados, ¿no? Es, por ejemplo, los encerados. ¿Qué es un encerado? Pues un encerado era un papel con aceite que suplía una ventana. Se le ponía ceras a los aceites y ceras a un papel y fungía de cristal prácticamente, dejaba pasar ciertas luces. Pero ese documento, que es uno más entre miles, que curiosamente le faltan fragmentos por otra parte, porque todo lo que firmara Palafox, quien organizó antiguamente el museo, el archivo, todas las firmas de Palafox se arrancaron. Y se arrancaban para regalarla porque era beato. Eran como una especie como de joya. Entonces, muchos documentos nos falta leerlos porque le han arrancado las firmas de Palafox. Y pasa también con estos, ¿eh? Pero aquí hay un caso muy, muy, muy bonito, que es que Palafox, y lo está diciendo él, le está pidiendo que más mandó el obispo, mi señor, cuidase que diere de color todas las cruces de la consagración en las cuales gasté lo siguiente. Las cruces de consagración es para el rito que se debe cumplir para consagrar, hacer un espacio, un edificio nuevo, hacerlo religioso, que esté bendito, es todo el programa de actos para que ese edificio consiga ese nivel. Y entre ellos ustedes entran a las iglesias y verán unas cruces en las paredes alrededor de toda la iglesia, en el interior, por supuesto, que es un rito que se hace ciertos rezos, se pone agua bendita, y en esas cruces que tienen que estar labradas en la piedra, en esas cruces lo que vamos a encontrar, es esto, ¿no? Y dice García Ferrer mandándole las facturas al obispo. Primeramente, una libra de bermellón, 2 libras de aceite de chía, monta 5 pesos y 6 reales. Si el papel con el que surge toda esta idea, uno de los principales junto con la batea y la detección en la batea, si ese papel es lo que nos ha dado a nosotros hoy, nos ha reunido ese papel de 1800 de la pintura de Correa, del amado Correa, de mi profesora Elisa Vargas Lugo, ahora nos hemos ido 50 años más tarde, o antes, a 1649, que es esta cuenta. Y lo está haciendo un español recién llegado, apenas 5 años está utilizando la chía. Eso nos lleva a perseguir, porque las cruces sí están, son esas cajas. Detrás de esas cajas están las cruces. Aquí pueden verlo. Ha sido muy complicado llegar a estos momentos de poder intervenir en la catedral y abrir. Pero cuando abres la caja, te pones nervioso porque ahí están las cruces de... Lo que te das cuenta es que hay un cristal. Porque esas cajas son del siglo XVIII. Y hay un cristal y ahí están las cruces rojas, como esperábamos con el Bermellón. Pero no se pueden acceder. Entonces buscamos algunas porque están clavadas al muro con clavos de forja. Es imposible sacarlas sin deteriorarlas. Pero siempre hay algún pero. Y a los pies de la iglesia están estos paneles. Yo soy historiador del arte, pero también soy restaurador. Y desarmador. Sin tener problemas llegamos a esa que le hicieron el panel para, ven ustedes el papel, eso lo hicieron para proteger la cruz porque se les desvió de cuando le hicieron toda la renovación les quedaba mal colocada y entonces lo que hicieron fue poner el panel para orientar todo a la misma altura, claro, y protegerla. Y ahí tenemos la chía, la famosa chía, pero cuando miras te dice, esto no es chía y esto no es bermellón. Cuando tienes la experiencia, estás descubriendo, dice, esto es carmín, el rojo mexicano. Eso es un repinte, después de 3 años para conseguir llegar a esto. Pero uno que tiene un poquito de ojo, lo que hace, dice, ahí está Ferrer. Entonces, a través de un deterioro, hicimos un escarbado con el permiso de la iglesia y de los responsables, y ahí debajo, en ese pequeño deterioro a 10 metros, se ve el rojo del bermellón. Esto sí es bermellón, con posiblemente chía, de la que sacamos una prueba. Pero si Ferrer está trabajando eso, y seguimos a Ferrer, y aquí les presento otro documento roto, ahí estaría la firma del doctor. Ven que está el doctor, ese es Palafox, alguien se la llevó. Pero aquí está Ferrer dirigiendo otros trabajos. ¿Y qué trabajo? No es cualquier trabajo. Es el trabajo concretamente de encargar a un platero que lleve lo que escribe Palafox, el santo, el beato, para colocarlo en el retablo mayor. Y se lo pide al platero. Y el platero lo hace. Y eso lo conservamos todavía en la catedral. Y eso nos lleva a pensar, si está pintando en pared con chía y bermellón, ¿con qué pintan aquellos que trabajan con él? Un platero que no pinta, pero está labrando piedra y está haciendo palabras. ¿Con qué? Porque se llama Lechuga, ahí tienen la firma. Estuvo tan orgulloso de su trabajo que hasta lo firmó. Piensen ustedes si se le rompe el panel en la última letra. Eso nos está diciendo otros lugares donde podemos encontrar la chía. Porque son también de la misma época. Como de la misma época es este pequeño fragmento que queda del antiguo retablo mayor, que se va reformando en el siglo XIX. Y ahí encontramos también el rojo y el verde. Ahí buscamos. Pero, para concluir, tenemos más preguntas. Hemos retrasado en el arte virreinal más de 50 años. Hemos mirado el arte virreinal desde otros puntos de vista. Hemos buscado preguntar y ahora tenemos unos fragmentos de color. Unos fragmentos de color que se vuelven maravillosas joyas para la historia cuando lleguen a la mano de todos los profesionales con los que hemos visto hoy. Pero, ¿qué hacemos sobre esos? Desde la historia del arte, lo que nos preguntamos ahora ¿es la chía mejor que la linaza para trabajar en otros materiales que no sean el lienzo? Muchísimas gracias.
Thank you very much, Pablo, and thank you to the other speakers. I'd like to invite all of you to take a seat at the front of the room, and I'm sure there will be plenty of questions from the room. Okay, there must be questions either from the room or among the three of you. Monica? Monica, I knew you would have a question. How did I know this?
So, I don't know whether you can answer this, Gerardo, but in your studies, did you find anything that would explain the current complaint that we heard from artisans when we were in Mexico, that even with this return to old techniques, they're still getting delamination. And I wondered, since you have gone through the various mixtures, whether there was anything just that would give you a hint as to what has caused this delamination.
That's perhaps the most difficult question ever. I just have guesses. All right, I just have guesses. And so that the question is whether the aje delaminates less than the chia oil gypsum, right? Because apparently that's the perception we have, that the aje has presents less delamination with time than the technology of chia, gypsum, kaolin. And so let's assume that indeed that's the case. And what is the cause for that? And I think it's a matter of contractions. If the oil that they are applying has already been treated by boiling it with lead and ajo and whatever, it speeds up the drying of the oil. But if you add too much oil, and you can see how generous they are when they prepare the koshle, which is this creamy thing with the kaolin. If you add too much oil, and then you don't subtract as much oil that the one you are adding, and you're already using oil that has been prepared to dry fast, that is going to create deformations in the surface of the oil, because the oil is going to deform. It's going to create like all these kind of like burrows, micro burrows, right? So that's one possibility. The other possibility is that if they are overcooking the gypsum rock, let's assume that they are not buying industrial gypsum, because that would be very bad. Because I don't think that the mucilage is going to provide enough water to actually revert the process from hemihydrated to rehydrated. But let's assume that they are doing everything fine. But if the rock is overcooked, then we're going to have there some kind of like gypsum that wants water, but it cannot get it because it's going to be sealed by the polymer. So that is going to be creating some kind of like stresses that I don't understand, but I leave them for you to think. The last thing that I think can be happening is the wood itself, because a lot of the modern delamination that I am observing is mostly on wooden furniture or wooden objects, not on the girts itself. And so one of the rules is that to avoid woods that have excess of resin. And since most of them are using today pine, that could be a cause. But the other cause that I think is more important is not even the resin in the pine, is that because they have such amount of production, because especially Olinala is a very successful center, that the wood is not being let dry for enough time. So these are just guesses. So I'm not saying that these are the causes. I'm just guessing from what I have seen and the processes I have seen. So I would just leave those ones there for you to think.
Yes, Hilda.
Hello, Marta Turok from Mexico. For Gerardo, I was very interested in your map, tu mapa, with the archaeological finds. Do you know about the what's it called, jícara from La Cueva del Gallo, 500 BC, with two things that are important: the rayado technique from Olinalá and traces of color, and we're missing. They haven't done enough studies to see if with the color there is also chia. It's in the, I don't know if they sent it back to Morelos. It's the Cueva del Gallo is in Morelos, or if it's in Mexico City. 500 BC, which would make it 2,500. No,
That would be
3,000
awesome.
years.
Just give me the content and I will go tomorrow.
Mi gusto. I want to make a comment on the first session when they talked about smell, odor, not smell, but odor. When we did our project 92, 94, with bringing the masters together and having them analyze their materials and trying to find out delamination, it all started with why is the lacquer delaminating? But anyway, so we were contacted by an agronomist from one of the universities in Jalisco, in Acatic, Jalisco, and he said, "Oh, I'm making chia oil. And I would like to see if the artisans of lacquer are interested in buying it because I want to use it. I'm thinking of using it in painting as an adhesive." So he gave us a few litros and we passed it around to the artisans of Guerrero, Chiapas, and Michoacán. And one of the first things that Chico Coronel from Olinalá said, "Pero no huele, it doesn't smell. Why doesn't it smell?" So we go back to him and we ask him, "What's your technique to process it?" He says, "I'm using cold press." And as everybody has heard today, you toast it first before you extract it. So then we said, "Well, try it anyway." And, you know, this person wants to know. So they tried and they said it lacks viscosity, which would be the mucilage, you know? So there were a series of things that just didn't make it convenient for the ones who were doing the most traditional lacquer work to incorporate it. So we told the researcher, we said, "Well, they're a little bit complaining that it doesn't smell." And he took offense. He says, "I'm not altering, no lo estoy alterando." You know, so it didn't work out. At least it wasn't used by the traditional lacquer people. And he had to look for other clients. But talking about the smell, it's an important aspect for authenticity, and probably not just the authenticity, but that the process is being done correctly. Just wanted to bring it out.
I don't want to be, but I have another thing is that we work also with Maya Blue, and the thing is like, how did they know what clay to use, right? Because it's a very special clay, and it's like, how do they select it? And we were making experiments with all different clays, and the students were trying to like pretty much all the possible clays you can imagine. We got them, and the students were like only with their fingers and not knowing what it was, but they have been given the touch first of the real Paligorskite, and they went through all the samples not knowing, and actually used with their fingers decided which one was the real Paligorskite. So which means that our senses are there, and that's why we decided to choose those materials, because they have properties that only with our fingers, our eyes, and we can decide if they are good. So we have our honest perspective.
One more question. Thank you. I have two questions, actually, very brief. One is for Gerardo. I was interested in the map that you showed at the beginning. And since the term sisa is something that we use also for gilding, I was wondering if you could clarify the term sisa related to age and to chia? If is there any difference? And my second question, sorry, and I just
I didn't understand. The sisa used for what?
for gilding, gold, sisa de dorar. And what is what you understand of sisa of faje and sisa of chia?
At least as we know it for the case of the lacquer, sisa means that you're combining at least one oil, pretty much linseed oil, with aje. And and that's the technique that is used mostly in
But not dryers, not
Michoacán.
pigments.
Not
dryers
no,
or
no,
not
no dryers.
pigments.
It's only
Okay.
it's only the oil and the aje.
Thank you.
And I have a belief, is because I'm not sure I'm right. The question is if the sisa was used in the pre-Columbian period, or if the sisa is something that was created as a need of the colonial period, especially because of the introduction of the linseed oil. Because as you saw, the linseed oil has very different ways to polymerize, and also if it is pure linseed oil, it dries. I'm waiting still for something that I let drying. It's month and it's still liquid, so it takes long to dry. But if then you add some of the lead or other things to make it a fast dryer thing, then you have other problems. But I think that the aje reduces the time for the linseed oil to dry. That's my guess, but we have the scientists here. They can decide.
And very brief for Pablo Madrol. Pablo, ¿tú has tenido alguna noticia sobre el uso de chia oil en pintura mural? Porque, claro, esto abre todo un universo de posibilidades para la pintura mural. Sabemos que el fresco era poco utilizado. ¿Algún artículo, alguna noticia, algún análisis?
No.
Gracias.
Bueno, ¿sí? ¿Se oye? Tengo buena voz. Vista no, pero voz tampoco, ni asiento. Ni asiento. Sí, ya está roto. Nosotros, la compañera Elsa Arroyo, con los sismos, el sismo del 17, en 2017, tiró muchos templos y afectó a una zona del sur de la Ciudad de México, con muchos de los conventos del siglo XVI, donde cayeron parte de sus pinturas murales. Además son de los grandes conventos de la ruta de conventos históricos protegidos por la UNESCO, y ahí ella misma, la doctora Arroyo, desarrolló toda una metodología para protección y estudio de esos, y la pueden encontrar en la página del instituto, se puede descargar incluso gratuitamente, y ahí han aparecido muchos materiales, hay infinidad queda mucha materia, pero la realidad, porque se ha guardado, la restauración no abarcó todos los fragmentos, pero no está apareciendo, han aparecido otros materiales, por ejemplo la obsidiana. La obsidiana está apareciendo en la pintura mural mexicana, ¿no? del 16, pero no está apareciendo la chía como tal, pero es que la chía sigue siendo nuestro quebradero de cabeza, es un aglutinante. Sí estamos encontrando nopal, sí estamos encontrando agaves, sí estamos encontrando, pero la chía en la pintura del 16, la mural del 16 no. Lo que sí, que es un poco también lo que estaba yo trayendo a Ferrer, lo que sí es que puede ser que la encontremos en la pintura de lienzo anterior a Correa, ¿no? Que es como nuestro punto de partida con el documento encontrado en el AGN. Pero hasta ahora, y que recuerde yo de los textos que he leído también de Lina, informes, no aparece nada extraño en ese sentido. Habrá que buscar. También es que no la sabemos identificar. Ahora nuestros compañeros de hoy nos han dado la varita mágica, ¿no? Lo malo es que a uno no se le da la magia. Gracias.
Okay, thank you very much. Do we have time for a short break before the final session?
Short break, but we're a little bit over time, so we'll start again in about 10 minutes.
Let's take 10 minutes.
Restrooms are just diagonal across the hall there, and please
Hi. Well, welcome everyone to our third session of the day, the third and last. I think it's a really special session. We have a great talk with Claudia Garza, and then a special demonstration of lacquerware from Uruapan, Michoacán, in Mexico. So let me first introduce our first speaker, Claudia Garza. Sorry, guys. She graduated in art conservation from the National School of Conservation of Mexico. She's a PhD student in art history from UNAM in the field of technical art history. She's a member of the Vice-Regal Sculptor Seminar. She was head of the conservation department of Museo del Carmen. She was painting conservator at the Museo Nacional del Virreinato. In addition, she has restored paintings from Museo Vizcaínas and Museo Franz Mayer, and she has also restored Vice-Regal devotional artwork. Her contribution in conferences and symposiums have been on the intervention of lightweight sculpture and painting on campus. So, welcome, Claudia.
Good afternoon. Good afternoon. It is a great pleasure to be with you this afternoon, sharing with great professionals as we have approached the conclusion of this symposium, which has proven to be profoundly enriching when we have deepened into the use of such an important material as chia oil. I extend my gratitude to the Metropolitan Museum of Art for the invitation and for promoting this type of research and events. In particular, special thanks to José Luis Lasarte and Julie and all the museum team that made this meeting possible. With the arrival of European artists in Mexican territory during the early years of the Viceroyalty of New Spain, works began to be created. They exemplified the fusion of diverse cultural influences, such as mural paintings, featherwork, codices, paintings, and sculptures, among others. Advancements in research conducted by archaeologists, historians, art historians, and conservators have enabled us to trace the use of materials native to the Americas. Although I am aware that we are at a symposium on chia as a binder, my presentation will focus on the supports, as this is what I have studied the most as an art conservator. So sorry, I will not speak about chia. I will present 3 cases that respond to the effectiveness achieved by using local elements, particularly organic materials used as supports for both paintings and sculptures. This includes maize stalks, amate paper, and various goods such as colorín, oyamel, and ayacahuite pine. These materials were used in conjunction with those imported from Europe, facilitated by trade relations that play a crucial role in the development of these artistic representations. Conversely, the 4th case corresponds to what happened with materials whose use was dependent on the importation of European products such as linen, which was not cultivated in American territories during the Viceroyalty. As a conservator, I have been involved in the study of the techniques and materials of New Spanish art, with a focus on their preservation. My work with Catholic Church in central Mexico has afforded me access to significant works requiring restoration and study. Among them, a large number of images are still in cult and present materials characteristics that have interested me. I would like to thank Dr. Pablo Amador, who just shared a very rich presentation a few moments ago, for introducing me to the irrevocable path of materiality studies, and for reminding me of the principle that restoration represents the methodological moment for in-depth examination of artworks, grounded in the dual polarity articulated by Cesare Brandi restoration theory. The professional field of conservation in Mexico is associated with public government institutions, such as the Ministry of Culture, the National Institute of Anthropology and History, the National Institute of Fine Arts, and various universities, such as UNAM, among others. Additionally, my work takes place in the private sector, where I manage projects and execute restoration both in situ and in the studio, primarily with financial resources from private individuals. In this context, access to studies on materiality in Mexico is often restricted to institutions that may lack the requisite equipment for such inquiries. While the UNAM has made significant strides in the study of materiality in New Spanish art, much remains to be addressed. The depending of topics such as the identification of binders has not received adequate attention. This is why I believe that this project, promoted by the Met, will yield substantial information regarding the artistic methodologies of New Spanish artists. The first case I will present is that of lightweight sculptures made with maize stalks. While this topic has been widely explored, and Pablo Amador just shared something about that, it has not been examined in depth within the context of Mexican academia. Currently, I am studying my PhD, and my research focuses on this particular type of sculptures made in the production center of Mexico City during the first century of the Viceroyalty. It has been widely acknowledge that the provenance of these images is Michoacan. However, thanks to Dr. Pablo Amador investigations, the existence of another production center in Mexico City has been identified, from which over 500 sculptures has been preserved to the present day. Lightweight maize sculptures are, alongside the architecture, the most enduring artistic testimonies of New Spanish art in its initial century. From the mid-16th century until nearly 1620s, these artworks were sent to Spain and other regions of Europe, commissioned by historically significant individuals and for prominent locations. The documentation preserved regarding these commissions has facilitated both geographic and chronological context of the images. The sculptures were constructed using material endemic from Mexican territory. The specific case I will present is that of El Señor de las Misericordias from Tlanepantla Cathedral, a lightweight sculpture composed of maize stalks created between 1570 and 1580. This sculpture shares characteristics with effigies preserved in Spain, such as the Santo Cristo de Zacatecas from Montilla, Córdoba, which suggests a potential time frame for the execution. The Christ measuring 88 inches in height, the crucifix from Tlanepantla weighs merely 18 pounds, reflecting one of the objectives of this likeness, to serve as processional sculptures. The great number of these artworks preserves attest to the stability and considerable devotional significance they have maintained, some of them for over 400 years. Sorry. Through X-ray examinations, along with knowledge and experience to identify it, a structure was revealed consisting of a mold craft from a mate paper, to which stalks of maize were adhered. These stalks were grounded, bounded with glue and other adhesives, and the maize paste was used to complete the sculptural form. The feet and hands and occasional internal pins were carved from coloring wood. A thin layer of a mate paper was applied over the paste that served to homogenize the surface. Upon the final layer of paper, the preparatory layers was applied, followed by the polychromies. The conservation treatments of the sculptures enable us to restore values that had been compromised. For the restoration, it is important to use the same materials so that they have behaved Similarly, and to prevent differential tensions. As you can see in the slide, some of the most common deteriorations are holes and losses of volumes. Thus, to remedy this damage, it is essential to follow the technique and depending on the stability of the materials, use the same ones as the originals. Once the maze support was done, we proceed with the processes of filling and retouching, placing a special emphasis on the polychroming strata. I will now continue to present the second case, is that of El Señor del Buen Despacho, a sculpture preserved in the parish of the same name in Mexico City. Formally, the crucifix has elements of 16th century sculptors, such as the disposition and shape of the bird. However, the erratic nature and little naturalism of its body correspond to those created in 17th century. Thus, I placed its creation chronologically in the middle of that century. The restoration was undertaken this year, allowing for deeper investigation into its manufacturing technique. The figure is similarly lightweight but materially distinct from those made with maize stalks. It was entirely carved from several sections of coloring wood, joined with glue, and has two hollowed areas. The head and the chest. Colorin is a soft wood, lightweight, from the tree Erythrina colaroides, an endemic species of America widely used for crafts and artworks. The Christ measures 75 inches in height and weighs 33 pounds. While it retains a degree of lightness, its weight is not as reduced as the maize sculptures. The crucifix had deteriorations that compromised both its aesthetic value and material stability, including a polychrome made in 18th century. The Christ also had undergone excessive cleaning, which causes losses on the surface and exposed the wood in certain areas. Furthermore, the church is located between heavily trafficked avenues. Thus, the entire polychromy surface was covered with a thick layer of dust and pollutants, which gave it a grayish tone. After the respective observation, diagnosis, and studies carried out, particularly X-ray and further laboratory results that we are expecting, restoration was performed, which sought to eliminate all dirt and contaminants, as well as to retouch all areas using glazes. It places it within art history, rendering it a significant reference for researchers of viceregal art and marking a transitional point towards 18th century sculpture. The preceding Christ are merely two examples of local materials employed as supports in viceregal sculpture. However, it is well known that altarpieces, other polychrome sculptures, and paintings on wood were also crafted using local woods, which I will discuss next. Although painting on canvas became popular in New Spain in the early 17th century, wooden panel paintings was used the most in the 16th century and remained so until the mid-17th century. Some examples have even been found in 18th century, particularly in altarpieces. The following example pertains to a series of paintings from the second quarter of 17th century. These 12 paintings on wood depict the lives of Saint Bonaventure and Saint John the Baptist and are located in their respective altarpieces in Tlalnepantla Cathedral. As Jonathan Brown notes, the new Spanish painting of this period exhibit particular characteristic that corresponds to inherited pictorial tradition from Europe, while simultaneously incorporate innovative techniques. New Spanish painting can be seen as leading area, along with architectures, in studies of viceregal plastic arts, to which analyses of materiality have been added in recent decades. In addition to its collaborative interdisciplinary efforts among institutions, including international partnerships exemplified by the Met, the field of materiality has emerged as a new pillar of research. Although this approach has long been established in international plastic arts, it has rapidly achieved significant heights within viceregal art. Due to the variety of goods available in New Spain, there was no necessity for long-distance transport for altarpieces and panel paintings. Therefore, we can deduce that the woods used in these paintings were sourced from nearby forests. It is particularly noteworthy to emphasize the importance of wood sections, and as its quality is evidenced by the stability resulting from proper selection, cutting, and drying. The wood species chosen for the panels is a yacuahuite pine, while oyamel was used for the crossbars. Biological studies of woods from panels made by Alejandra Quintanar identify pine as one of the most used woods, making it unsurprisingly to find this species in the works from Tlanepantla. Rather, it confirms affiliation with a workshop that effectively used local resources and operate in accordance with contemporary production. In addition to the woods, agave fibers were also employed, adhered with glue to provide stability to the panels. For the next case, I will discuss a topic I have addressed on other occasions, which is interesting because it continued to discuss of supports, this time focusing on linen canvases. This concerned to an apostolate from the parish church of San Francisco de Asís Atizapán, created in the mid-18th century. Analyzing this collection of paintings attributed to a single anonymous author proved particularly interesting as it provides exceptional documentation regarding the technical and material solutions, specifically concerning the textile support of a mid-18th century viceregal artist. As some researchers have pointed out, there is a lack of information about the practice of painters in documents, so approaching the subject through material analysis is the best way to expand knowledge of New Spanish art technique. Departing from the observation and analysis of the paintings, I would like to emphasize that we found restricted use of the linen. Also, 12 of the paintings have regular patterns of perforations and marks made with ferrogallic ink that confirm the use of canvases. In this context, after registering all these characteristics and trying to find information, we realized that the analysis of canvas had not been granted the importance it deserves. The information that we can obtain from the analysis of these canvases is very important because it also gives us information about origin, circulation, and the loom that was used to make the textiles. The material exchange between Spain and New Spain was fundamental, particularly concerning the importation and exportation of textiles from Spain and other parts of Europe to New Spain, as well as other materials such as pigments, lacquers, oils, and brushes. In relation to the topic, the case of the series of these paintings is noteworthy. The specific findings, patterns, and perforations and marks made with ferrogallic inks provided insight into the acquisition of fabric by the artist. These marks correspond to the cargadores. The cargadores were people whose work was to carry merchandise on ships from one continent to another. The products were carried in bales or bundles of linen that were numbered and marked with their initials. The canvases of the paintings were made from such linen bales that the artist had bought to paint over them. Furthermore, in the Thyssen-Bornemisza Museum, the painting The Custom Yard that displayed some traders with their merchandise bundles in bales, that have similar marks. No? You can see there. Additional paintings and ex votos documented this practice. In conclusion, I would like to mention that the field of restoration and conservation in Mexico has much to contribute to technical art history, and that it is in places like this symposium that we must share information in addition to the necessary publication of results. I would like to raise an important question as a final reflection. Did some of these materials serve as supports for polychromies or pictorial layers using chia oil? Thanks to the ongoing efforts by the MET team, we hope to explore such questions further in the future. Thank you.
Thank you, Claudia, so much. Let me introduce now Martina Navarro González, Tomás de Saint-Faux, and Erandine de Saint-Faux, our lacquerware artists who are going to do a demonstration of their craft. So Martina Navarro and her family lead a workshop dedicated to creating and promoting exquisite handcrafted wooden artworks. Their workshop is based in Uruapan, Michoacán, a region renowned for its rich tradition of artisanal craftsmanship. Building on this legacy, Martina began her journey in 1986, producing small quantities of lacquered wooden pieces while refining her skills through competitions and meticulous practice. By 1984, she expanded production and gained recognition for her mastery of maque, an ancient decorative technique that involves applying natural pigments and drying oils spread evenly using the palm of the hand to create lustrous and terse surfaces. The family workshop specializes in creating intricate lacquerware designs on wood and guaje, a type of hollow bread-based gourd, as well as finely turned wooden pieces. Each item is crafted using traditional pre-Hispanic techniques and natural high-quality materials, preserving a centuries-old heritage. Their work is defined by a blend of respect for these time-honored techniques and an innovative approach to design, resulting in a uniquely beautiful trays, words, and boxes celebrated worldwide for their quality, functionality, and artistic elegance. And you can see some of those pieces displayed already. So help me welcome Martina, Tomás, and Erandine, please.
Buenas tardes. Nosotros vamos a hacer una pequeña demostración de las diferentes etapas del proceso de producción. Antes de que ellos empiecen, voy a hacer una pequeña descripción de los materiales que nosotros utilizamos. La principal, la base de nuestro trabajo es la tierra. A la tierra le vamos a ir agregando pigmentos, lo que le va a dar un color diferente a cada... que vamos a obtener diferentes colores, una gama diferente de colores. Principalmente en Uruapan utilizamos una tierra que se llama charanda. Es una tierra rojiza y que la palabra proviene del purépecha, que significa rojo, que es chalapit, que significa rojo en purépecha. Y esta tierra, cuando al aplicarla, te va a dar un color rojizo. Entonces, a esta, por ejemplo, el negro que vamos a utilizar el día de hoy, vamos a ponerle el hollín. El hollín, pues, es lo que sale de la chimenea y que es lo que nos va a dar un color, un tono rojo. Y para poderlo aplicar, vamos a utilizar el aceite. El aceite, nosotros utilizamos actualmente aceite de linaza, ya no se utiliza el aceite de chía por cuestiones de costo y por cuestiones de que no se consigue tan fácilmente. Nos ha funcionado, a nosotros nos ha dado una muy buena calidad, y es lo que nosotros utilizamos. Antes de utilizar el aceite, lo vamos a preparar. Le vamos a poner que es el aje, que es la grasa del animal que se mencionó ya anterior varias veces y que es la grasita que al hervirse suelta una grasa y esa grasa es la que a nosotros nos sirve como secante. Y para nosotros es súper importante porque es lo que va a hacer que sequen más rápido las piezas y que le dé una durabilidad. Y a este aceite le vamos a echar el ocote y ajo. Entonces ya cuando preparamos esto, ya está listo el aceite para nosotros poder trabajar. Entonces la primera etapa que va a hacer ahorita mi mamá va a ser el fondeado, que es la base de la pieza. La madera que nosotros utilizamos siempre va a ser una madera que no tenga resina. Se utiliza principalmente el aile. El aile es una madera que se encontraba en la región de Uruapan o en la región purépecha muy fácilmente. Actualmente ya no hay tanto aile y también las personas que producen las bateas, ahorita nada más tenemos una sola persona. La batea es el plato de madera. Lo que nosotros le llamamos la batea es el plato de madera. Y principalmente, pues, ahorita ocupamos el aile para las bateas pequeñas. Para las bateas grandes o los platones grandes, como de un 70 centímetros, un metro de diámetro, utilizamos la parota, que lo hacen es de la costa de Michoacán. Entonces, mi mamá va a empezar ahorita a hacer el fondeado.
Buenas tardes. Les voy a hacer una demostración cómo nosotros trabajamos esta artesanía. Como lo dijo mi hija, ya creo que ya explicó casi todo. Yo, para volver a retomar esta artesanía, hace como 40 años que lo retomé. A lo mejor una nueva técnica o para mejorar, estuve en varios encuentros, 3 encuentros con la señora aquí presente, Marta Turok. Yo aproveché mucho porque nosotros, desde que comenzamos con mi esposo a trabajar esto, el objetivo era hacer investigaciones y hacer piezas utilitarias. Para eso nosotros teníamos que dejar los secantes industriales y el yeso. Comenzamos nosotros a hacer experimentos con las tierras del campo que nosotros mismos las íbamos a buscar. Y yo, con la experiencia que demostraron mis compañeros artesanos, pues yo traté de aprender y ver cómo los demás artesanos trabajaban. O sea, aprendimos, creo que de los 3 estados que hacemos el maque o laca, como se dice, Chiapa de Corzo, Guerrero y Michoacán. Entonces, pues aprendimos un poco de cada quien, tanto ellos como de nosotros como nosotros de ellos. Y ahorita lo que voy a hacer es demostrarles el fondeado, que el fondeado es Nosotros, como lo van a ver, no utilizamos pinceles. Pues enseguida voy a comenzar. Esto que voy a hacer ahorita es el fondeado, y la primera capa le decimos sisa.
En realidad lo que llamamos sisa nosotros es la mezcla de aceite con polvo. Que ahorita agarra un poco de aceite y un poco de polvo y lo mezcla sobre la batea. Es lo que llamamos sisa nosotros.
Mucha gente pensaría que le va a hacer no sé, alergia o que le van a doler las manos. No, al contrario. Al contrario, yo tengo 40 años más o menos trabajando en esto y mis manos están bien, están limpias, no tengo artritis, no están ásperas, mis manos están lisas. Y yo pienso que porque nosotros trabajamos solamente con los materiales muy naturales. El negro, les explico lo del negro. Esta preparación que hicimos con el negro es tierra natural, que es la tierra charanda, como le llamamos nosotros. Y se molió esa tierra con el hollín, el tizne que hay en las en la chimenea. Pero a este negro no solamente es tierra y negro. Le aumentamos, le ponemos un poco de azul de Prusia, que es un pigmento mineral. Porque si yo no utilizo el azul de Prusia, el negro va a salir gris. No va a ser un negro intenso. Entonces es así como trabajamos y es capa por capa.
Y después de poner aceite con tierra, lo expande bien y luego le pone una capa de polvo con el trapo, luego expande aceite puro.
Así como
lo expande con la palma de la mano. Todo se trabaja con la palma de la mano. Y ahí el chiste de alisarlo bien para que no se hagan bolas, que no se haga chino. Y para que pueda brillar bien tiene que estar muy liso.
Entonces tengo que trabajar muy bien, cuidar cómo estoy empezando desde el inicio para que mi trabajo quede bien. Este no tiene nada de barniz industrial para que brille, es brillo natural. Entonces yo tengo que trabajar bien desde el inicio. Entonces yo poco a poco le voy a ir apretando más, hacer esto, para que desde el inicio el trabajo comience bien. Bueno, como todo, ¿verdad? Si se comienza bien, va a terminar bien. Es muy agradable trabajar con las tierras naturales, sobre todo esta tierra que estoy utilizando es muy agradable. Se siente que es algo natural.
Bueno, la charanda, yo creo, cuando la fábrica de charanda empezó, le pusieron charanda porque realmente en Uruapan la tierra es rojiza y hay o sea, alrededor de Uruapan la tierra es charanda, y es muy rojiza también.
Y en realidad nos explicaba la hija del fundador de esa fábrica que le puso nombre de charanda a su bebida porque su fábrica la instaló en el cerro de la charanda. Hay un cerro ahí a la entrada de Uruapan que se llama La Charanda y ahí tenía su fábrica, entonces le puso el nombre de charanda a su bebida.
Sí, charanda viene de la palabra purépecha, charapiti. Y entonces, como la tierra es rojiza.
Sí, otra de las tierras que nosotros utilizamos es una tierra que buscamos cerca del lago de Pátzcuaro. Es al lado del lago de Pátzcuaro y es una tierra que es muy transparente, entonces esa nos permite dar colores claros. Esta roja no nos permite dar colores claros. Por ejemplo, si queremos hacer un amarillo vibrante, no podemos utilizar esta tierra porque tiene fierro, entonces es muy rojiza. Entonces, si queremos un azul brillante también no podemos utilizar esta. Entonces, esta la utilizamos para colores más oscuros, que van a ser negros, azules muy fuertes, o tal vez un amarillo ocre. Y la otra que es más transparente sí nos permite utilizar pues hacer el resto de los colores.
Entonces, no solamente pintamos o trabajamos la madera, también trabajamos el guaje. Trabajamos los guajes o bules, no sé cómo. Esta es una parte.
Es gold en inglés. Gold. ¿Es esto o esto? Esto es el fondo de una calabaza, le cortamos y se hace lo que llamamos jícara.
Y tradicionalmente estas piezas en Uruapan se utilizaban mucho para hacer obsequios o para llevar regalos, o para un bautizo, por ejemplo, ahí se llevaban los regalos, o cuando alguien se casaba en una boda, por ejemplo, a la novia era un gran honor cuando le regalaban una batea, o las familias mandaban a hacer sus piezas, sus bateas especiales con el sello de la familia, por ejemplo, pero antes era más, se utilizaba mucho para los obsequios. So, ahorita ya realmente no lo utilizamos para eso.
También hacían piezas más sencillas para, por ejemplo, sacar el grano de los costales, el maíz, o también hacían jícaras chiquitas para tomar atole o chocolate. Eran piezas útiles y, por ejemplo, era uno de los retos que teníamos nosotros porque en el siglo pasado hacían mucho maque, pero utilizan mucho yeso al lugar de tierra y no eran permeables. Con el agua se despinta o se descarapela. Lo que tratamos de hacer es buscar materiales adecuados para volver a hacer piezas impermeables y que duren más. Y si lo logramos, por ejemplo, ahí le pueden poner agua y no le pasa nada.
Ese era nuestro objetivo, llegar a hacer piezas utilitarias que se mojaran y que no les pasara nada. Y creo que hasta ahorita lo hemos logrado.
Ahorita lo que está haciendo mi mamá ya le llamamos nosotros emparejar, que le está quitando el exceso de material o de aceite y le está quitando todas estas partes que se van a ver rugosas o se van a o haya poros. Ahorita tal vez no se aprecia muy bien porque siempre en lo negro no se ve muy bien, pero lo que está haciendo es darle como que se vea más terso y más lo más liso posible. Después de eso le va a poner más polvo para lo que nosotros le llamamos sacarle brillo. Entonces, entre más polvo le ponga, se va a secar más rápido y va a brillar más. Entonces, eso hace que cuando nosotros lo pulamos, le vaya saliendo brillo. Y el brillo que ven es lo que al secarse lo vamos, va saliendo.
Hay una cosa que muchas veces hacemos el fondo en negro porque es más fácil combinar colores en fondo negro. Y mucha gente piensa que el maque siempre es en fondo negro y no es así. El maque se hace de muchos colores diferentes. A mediados del siglo pasado hacían muchos fondos de colores pasteles, en azul, en
grises,
verdes, en blanco también. Pero utilizamos mucho el negro porque es lo más fácil, por ocasión de facilidad.
Pero se ensucian más las manos, a veces hasta la cara.
Entonces, después de acabar el fondo, ahora le va a poner mucho polvo, muchas capas de polvo para que vaya secando. Y al día siguiente le volvemos a dar brillo con un trapo seco. Y así durante una semana, cada día dándole brillo para que esté brilloso así naturalmente sin poner barniz.
Alguien dijo que había que darle muchas repasadas con algodón para que secara, pues yo creo que es eso. Nosotros limpiamos ya después que ya yo terminé y que ya está un poco seco con el algodón para secarlo. El algodón como que le quita más la humedad, lo que...
Entonces, yo le dibujo. Mira,
sí. Después de esto se deja secar entre unos 6 y 8 días.
Ya
para
cuando
poder...
esté seco la batea, lo dibujamos. Por ejemplo, ahorita lo dibujo con un lápiz de cera que me permite está bien complicado eso. Con el lápiz, si me confundo, lo puedo borrar y lo hago otra vez. ¿Sí? Hay artesanos que dibujan directamente así con la aguja.
Con esto o con una aguja.
Sí. Entonces, yo prefiero así con el...
Sí, porque si te equivocas con la aguja, por ejemplo, ya no hay vuelta atrás. Ya no podemos borrar.
Entonces, una vez que esté dibujado, con un cuchillo lo marco. Bueno, aquí.
Ahorita lo que está haciendo, bueno, lo está marcando y lo va a rayar y va a quedar un huequito. Ese huequito, vamos a quitar lo que ya pusimos. Entonces, se raya y se va quitando. Entonces, entre más tiempo pasa, más duro es y más difícil es quitarlo. Entonces, siempre preferimos trabajar que no tengan más de 4 o 5 meses las piezas porque ya después es muy duro de quitar, te cuesta más trabajo. Entonces, ahorita, dejamos secar unos 15 días más o menos, o 8 días, o sea, el tiempo mínimo son 8 días, definitivamente ya hemos hecho el tiempo calculado y si es mínimo 8 días. Y si es húmedo, es más todavía porque la humedad hace que se tarde en secar, se tarda mucho más en secar. Entonces, en tiempo de secas es ideal porque se seca súper rápido. Entonces, nosotros, sí, aproximadamente después de 10 días más o menos, empezamos a trabajar la pieza y ponemos el siguiente color. Porque si no esperamos a que se seque, al momento de poner el siguiente color se mancha. Entonces, si ponemos amarillo, por ejemplo, el amarillo va a quedar como gris, si es negro o se va manchando, ya no queda un color muy limpio. Entonces, sí es importante dejar de secar. Entonces, nos ha pasado que la gente cuando toma cursos se desespera porque ya quiere poner el siguiente color y no puedes, tienes que a fuerzas dejar secar.
Y esta, por ejemplo, la pintó Martina como fue al fin del mes pasado.
Cuando di la demostración en
Xcaret.
Xcaret, sí.
Pues fue como el 25 de septiembre y ya está dura. Ya me da trabajo.
Nos ha pasado que a veces se nos cae una batea ya terminada y más bien se revienta la madera y al maque, a la pintura no le ha pasado nada. Quiere decir que se adherió muy fuerte, muy, muy fuerte. Yo pienso que hacíamos una reflexión con mi hija estos días que tenemos las uñas muy, muy duras. Y yo pienso que es el aje, la resina de pino y el ajo de cocinar. Pensamos que todo eso nos ayuda. Y por lo tanto, pues hace muy, muy, muy sólido al maque. Por eso, cuando mojamos las piezas, no les pasa nada. Mi hijo, que bueno, es un joven que pues toman mezcal y esas cosas. Entonces, sus amigos le empezaron a decir, oye, pues haznos unas copitas de lo que haces de tu trabajo, en pedacitos de guaje. Entonces él empezó a hacer vasitos así y han tomado mezcal allí y pues bien, no le ha pasado nada. Le siguen mandando a hacer. Esto que estoy haciendo ahorita ya es lo último para que quede lisito y que ya empiece a brillar. No igual, pero que ya. Y mucha gente piensa que nos preguntan que de cuál laca utilizamos para que brille. Pues no, ninguna laca. Esto pues es fácil y para mí hasta es incómodo porque yo estoy acostumbrada a pintar piezas grandes.
Sí, la pieza más grande que hemos, bueno, de los platos, de las bateas, la más grande que hemos pintado es de 1,10 metros. Está bien grande. Y pues ya todo el día se han preguntado por qué la chía, por qué la linaza. Y pues veo que hay muchas razones, ¿no? Que la linaza a lo mejor, la chía a lo mejor seca más rápido o seca mejor, o la linaza tiene otras ventajas y todo. Pero nosotros tenemos yo creo que para tomar la decisión un punto crucial es cómo la conseguimos y a qué precio. Y mucha gente utiliza la linaza porque es lo más fácil de conseguir.
Y barato también.
Y como decía el señor, que en el siglo XVI, XVII encontraban la chía en la tienda y nosotros no. Y nosotros como artesanos no podemos hacer todo, hacer el aceite, preparar los pigmentos, cultivar la chía, cultivar ¿cómo se llama? La grana cochilla, no se puede. O eres agricultor o eres artesano, no puedes hacer todo, el tiempo no nos alcanza. Entonces uno tiene que escoger. Por ejemplo, yo soy ebanista, pero también soy maquillador, pero ya no puedo yo hacer muebles porque no me da tiempo. Entonces prefiero comprar la batea y pintarla. Entonces, a veces la razón es de utilidad o de práctica, ¿no?
Papi.
Ya casi brilla.
Entonces, ya lo que quité, lo voy a incrustar. Entonces es igual, le pongo aceite con pigmento, con polvo y lo incrusto bien en la parte que quité.
Se le olvida.
Pero no veo. Y con el algodón le pongo polvo. Y la razón por la cual debemos de dejar secar bien el fondo es para que el verde no se pegue en el azul.
Este proceso bueno, lo que está haciendo ahorita mi papá es lo que le llamamos el incrustado, como él lo dijo, y vamos a hacer por colores. Entonces ahorita él puso el verde ya que está seco, vamos a dejar secar unos 8 días, vamos a recortar las siguientes, el siguiente dibujo que querramos. Entonces, si es una flor roja, pues vamos a recortar todo lo que es rojo y vamos a aplicar el rojo. Dejamos secar el rojo y volvemos a aplicar el siguiente color. Entonces, entre más colores lleva, pues más tiempo de secado, más tiempo pues para tener la pieza terminada. Más o menos una pequeña pieza de 3 colores, 2 colores, 3 colores realmente son un mes, porque realmente si calculamos 8 días por cada color, más o menos es un mes. Ya que una vez está terminada bien ya hemos puesto todos los colores, vamos a hacer lo que le llamamos perfilado, que es ya lo último que hacemos, que es hacerle una línea blanca a las flores, y esa línea blanca va a dar más color y más movimiento a más luz a las bateas. Y algo que es muy específico de Uruapan y que diferencia de Chiapas y de Olinalá es que nosotros vamos a quitar para volver a rellenar. Eso sí es súper importante, que es algo que diferencia a los otros estados. Y se le llama maque y se le llama perfilado también por la línea blanca que le hacemos en todas. Les voy a no todavía porque va a quedar. Bueno, les iba a presentar la diferencia.
El perfil es la raya blanca. Es la raya blanca. Si gustan ver. Es lo que es para darle luz a la flor.
Y tenemos en el incrustado, por ejemplo, tenemos que dejarlo lo más, que no quede relieve, o sea, que no quede la mitad lleno, que tiene que quedar lo más uniforme con la base. Eso sí es importante, que no sientas ahí el bultito de que se volvió a llenar.
Más o menos. Ahí no lo hice muy bien, le queda con unos pedazos de azul. A ver, ¿te quieres hablar de
¿Listo?
Abajo?
A ver, deja.
Lo que en cuestión de decoración, la forma de dibujar y de incrustar nos limita mucho. No podemos hacer los efectos que tienen la laca de Pátzcuaro o de ¿cómo se llama? De Chaparricos o de Nalá, porque ellos lo hacen a pincel. Entonces, pueden poner un color sobre otro color al instante, entonces te hacen mezclas o degradados que nosotros no podemos hacer. Entonces, nosotros más bien trabajamos con plastas, así, colores unidos, es un poco más bien un trabajo de maquetería. A mí lo que aprendí en Francia de la maquetería y todo eso me ayudó mucho, porque es el mismo, como la misma mentalidad, la misma forma de pensar el dibujo.
Aquí les voy a presentar unas piezas que hemos hecho en el taller. Esta parte es lo que nosotros le llamamos maque tradicional. El maque tradicional es muy, bueno, lo definen porque siempre, entre comillas, ¿no? Dicen que para ser tradicional, tiene que ser de fondo negro y siempre tiene un centro, que es el centro, por ejemplo, en la batea más grande tienes el centro de pájaros y ramos de flores. Entonces siempre la mayor parte de estas bateas tienen un centro, una greca alrededor que va a dividir con el otro, con un conjunto de flores. Entonces casi la mayor parte siempre, como ven, tienen un centro, tienen la greca y tienen otro ramo de flores o una guía de flores alrededor. En la parte hay uno que tiene una batea que tiene filo amarillo. Esa pieza no está perfilada. Esa, si ven, tiene como un degradado rojo. Entonces, esa parte nosotros le llamamos manchado o sombreado. En las otras es perfilado porque si ven en todas las flores tienen una línea blanca.
Es el manchado, eso.
En esta también ven, por ejemplo, las amapolas, es el manchado, que es una variante de lo que se hace en maque, que es el perfilado y el manchado o sombreado, que le da otro efecto, que le da algo más realista a las flores. Y bueno, siempre se utilizaron flores que venían o que eran de la región, ¿no? Esto es un guaje, que es de rosas, es algo que nosotros también pues trabajamos la madera y el guaje. Entonces, esta es la técnica de sombreado y que es la parte más tradicional del maque. Esto son guajes que ya fue lo que nosotros ya en el taller, como que en la época que mis papás, pues una cosa ya de hacer más lo que tú tienes ganas y lo que se antoja, pues sí, tienes ganas. Y para vender también, que era una problemática que teníamos, empezamos a hacer cosas más sencillas. Y esto fue una de las, pues no de las primeras, pero sí fueron de los modelos o de los métodos que nos permitieron tener, que se vendían mucho, ¿no? Y que fue que empezamos a producir más piezas que no fueran las tradicionales. Y bueno, realmente creo que nunca hemos sido tan tradicionales y ha sido siempre muy cuestionado en el área del maque, entre los artesanos del maque. Entonces, pero estas sí han sido una de los modelos que más
una de las líneas que más nos ha funcionado, porque siendo la técnica original, pero con más sencillo, entonces más barato, y nos permitió sobrevivir con el maque. Como son piezas más baratas, de calidad, pero más baratas, entonces se vendían mejor y así pudimos seguir con el taller. Porque en realidad es un poco un problema de muchos artesanos. Siguen haciendo piezas tradicionales que son muy costosas y que no corresponden al gusto actual. Entonces no se venden. Si no se vende, yo no trabajo. Entonces, porque nadie trabaja por el arte, uno trabaja para comer.
Y estas piezas, bueno, estas piezas son en 3 semanas, ya están secas, ya podemos venderlas. En comparación a este baúl que nos llevó como 4 meses más o menos de producción. Entonces, esta pieza la hicimos para un concurso, pero pues entre comparación de una pieza que terminas en 3 semanas a esta que terminas en 4 meses, pues son trabajos, tiempos de producción muy diferentes y que, bueno, uno nos permite producir estas grandes piezas, ¿no? El maque actual, bueno, lo que nosotros ya hacemos más con la nueva generación, que ya somos mi hermano, Irepan, que es el que ahorita está trabajando con nosotros, pues yo y ahora mi sobrino, que es el que le estamos enseñando y que es la nueva, ya somos los nuevos en el taller. Estas son las piezas ya más contemporáneas que nosotros, la línea que más hemos trabajado. Eso fue un cráneo. Hicimos el experimento a ver si funcionaba en el cráneo y por el momento no se han quejado, entonces sí funciona.
Nadie se ha quejado.
Sí, y trabajamos más estilos más, mi hermano y yo, más geométricos y menos colores también. Estas fueron unas piezas. Esto es un guaje, el que tiene las líneas, bueno, el blanco con azul. Esa es un guaje que normalmente es vertical, pero nosotros lo pusimos horizontal. Le pusimos una base de madera para que pudiera pararse. Entonces, esto ha sido de las últimas piezas que hemos hecho. Mi hermano Irepan, que trabajó esa pieza que tiene rojo y negro, entonces hemos trabajado dibujos más sencillos. Y esta fue una pieza que hicimos para Design Week de México el año pasado. Y pues se inspiró mi papá de varias cosas. Hay un dibujo donde hay el barco con un pescador. Ese dibujo mi papá lo tomó de una batea de Diego Rivera que mandó a hacer en Uruapan. Esa que lo hicieron, él hizo el dibujo, el diseño y los artesanos de Uruapan lo hicieron. Hay el perro, que es el símbolo del, pues el guía que te lleva al inframundo. No veo qué más, el pájaro.
Hay la garza, que son un pájaro que hay mucho en el lago de Pátzcuaro, y hay el otro es un coyote, creo. Que son símbolos, pues, que yo retomé muchos símbolos de la cultura purépecha.
Sí.
Nosotros nos inspiramos, cosas así contemporáneas, pero también retomamos dibujos prehispánicos.
Y bueno, estas son piezas que ya más sencillas, que tienen 2 colores y también siempre es como una inspiración más de piezas prehispánicas o más geométricas. Hace unos años hicimos una colaboración con Abraham Cruz Villegas, que hizo una exposición en el Art Club de Chicago. Entonces, él nos pidió que trabajamos con él en diferentes piezas que teníamos el taller y que ya íbamos a tirar y que nosotros intervenimos. En la otra foto es una exposición, son 2 piezas que ganaron premio y una es de 80 centímetros, yo creo, y la otra de metro y la otra es de 70 centímetros y que hacen parte de la colección Banamex, que es una de las mejores, que tiene muy buena colección de arte popular. Y pues ahí vemos el contraste de lo que en la foto donde está la batea, vemos el contraste entre lo más tradicional y lo más pues lo más contemporáneo, ¿no? Que es como la copa negra que yo hice y la otra que es de mis papás, que hace parte de la colección Banamex, y fue una exposición que se hizo en la ciudad de Uruapan, en la fábrica de San Pedro, y que era una exposición sobre las piezas, el diseño y la artesanía, ¿no? Entonces ahí vemos las 2 diferencias, ¿no? Entre los polos opuestos de lo más contemporáneo y lo más tradicional.
La batea grande es de madera de parota y la copa son pedazos de bules de guaje que asamblamos para hacer una copa. ¿Y la otra foto?
La otra foto son unas piezas que hicimos para Ensamble Artesano, que es una fundación que trabajó con artesanos y diseñadores, y nosotros presentamos los 3 guajes, el rojo, amarillo y negro, y los otros negro y amarillo, que hicimos una colección especial para esta fundación que trabajamos con ellos. Y listo, es todo. Muchas gracias.
No sé si quieran preguntar.
Muchas gracias, Martina, Tomás y Erandi. Si quieren, pueden pasar a sentarse allá. Y también Claudia, si quieres, tenemos tiempo, unos minutitos para preguntas. Como 1 or 2. O sea.
Yo, mira, me traje eso.
If not, yo puedo hacer una también, si quieres. Si nadie tiene. Bueno, yo quería hacer, en lo que se acomodan, una pregunta a Claudia y uno a la familia Saint-Foll. Primero, Claudia, quería saber si tú nos presentaste muchos materiales locales y en tu opinión, ¿cuál es la importancia de incluir en una historia del arte global? O sea, ¿cuál sería tu reflexión de estar pensando en estos materiales que antes no habían sido considerados tal vez por una historia del arte enfocada en Europa o en Estados Unidos y que en la Nueva España fueron usados desde hace mucho tiempo, ¿no? Y para la familia, bueno, muchas gracias. Y para ustedes, ¿cómo la experiencia de, vimos en la mañana todos los científicos hablar sobre Lacas y sobre su trabajo y ustedes lo hacen en la práctica? Y me gustó mucho cómo ellos dijeron que no es hasta que ellos los ven hacerlo que entienden en realidad cómo funciona todo. Entonces, ¿cómo ustedes experimentan al revés, ver la parte científica de su trabajo? Y bueno, yo creo que esas.
Bueno, ¿se escucha? Sí, ¿verdad? Pues en realidad es como muy interesante el poder, o sea, sí han sido como tomadas por la historia del arte, pero en realidad no han sido del todo como puesta la atención en ese punto, ¿no? Y como la vinculación con lo europeo también, ¿no? Con las técnicas que vienen y que se apropian los materiales. Creo que esa es la parte que hace falta, ¿no? En donde hay una reflexión como más profunda, como de un trabajo en conjunto y que se había perdido, ¿no? O que se había visto como muy, muy separada. Entonces creo que va un poco por ese lado, ¿no?
Yo de los materiales, yo soy de Francia, entonces cuando llegué a México conocí el maque por mi esposa y me pareció muy interesante porque es una técnica excepcional. Utilizan materiales que pues, muy comunes, aceite y la vil tierra, y hacen sacan un producto extraordinario de muy buena calidad. En Francia la gente es muy exigente, es demasiado exigente y les gusta el maque. Entonces quiere decir que es una técnica de muy buena calidad. Y yo he visto, por ejemplo, en Uruapan, que la gente lo desprecia, no lo saben valorar. Y yo por eso me interesé mucho a eso y empujé a mi esposa para que lo siguiera haciendo, porque ella estudió enfermería y lo abandonó. Y pues yo la empujé a que lo hiciera, porque es una técnica que no se debe de perder.
Bueno, yo de mi punto de vista ya más, creo que más práctico, que soy una persona más práctica, para mí es algo importante escuchar todo lo que se ha dicho, ¿no? Porque es como que darle ciertas palabras a lo que ya estoy haciendo y es también una parte de la formación, ¿no? Porque como artesano, pues también tenemos que formarnos y es parte de la formación que yo estoy recibiendo para dar otro paso más adelante, ¿no? Como cualquier persona en un trabajo, pues sigue formándose, ¿no? Entonces, para mí esto es algo como parte de mi formación y de mi aprendizaje y darle tal vez no una justificación, pero sí algo decir, sí, lo que estoy haciendo va por el buen camino, tal vez, ¿no?
Pues yo cuando estaba escuchando y viendo tanta, lo que contienen los materiales, me dio un poco de risa porque yo dije, pues yo cuántos años tengo utilizando esos materiales y yo no sabía que yo estaba, sí, entonces dije, pero bueno, todo eso pues es muy bueno porque yo estoy bien, aparentemente sana, ¿verdad? Ya 40 años sin dejar de trabajar con las tierras y tienen hasta un ADN que Y yo dije, ¿cómo? Y bueno, pues para mí es muy importante hacer estas demostraciones. Me gusta mucho y estoy muy contenta que me hayan invitado, que hayan pensado en nosotros y que nos invitaron a venir aquí a demostrar lo que nosotros sabemos hacer, y es algo que para mí yo lo llevo dentro de mí, porque a veces pienso que soy la herencia o es la herencia de mis antepasados purépechas, porque ellos eran los que trabajaban ahí en el pueblo de Uruapan, que antes eran purépechas. Ellos trabajaban el maque, ellos pues hicieron muchas piezas que a veces aparecen todavía por ahí. Y bueno, pues cuando la llegada de los españoles los quitaron, los sacaron fuera de Uruapan, pero aún me imagino que se quedaron algunas personas ahí que lo siguieron haciendo. Y después, pues hubo más gentes, más personas que lo hicieron. En tiempo del volcán, cuando brotó el volcán de ahí, el Paricutín, que está cerca de Uruapan, hubo mucho auge, había muchos maquiadores, trabajaron mucho, pero después se vino en decadencia otra vez y ya no había quien, habían 2, 3 personas nada más que lo hacían, y ya mientras, pues ya llegamos nosotros. Y yo, una de las pocas personas que quedaban, yo platiqué mucho con él, un señor ya grande, y él me dijo muchas cosas, me contaba muchas cosas, pero para eso, yo cuando tenía 12 años, me enseñaron, no muy bien porque fue por poco tiempo. Yo estaba en un internado y allá fue donde yo tuve esa oportunidad de aprender y de saber lo que era el maque. Entonces, pues después de haber estudiado y trabajar y otras cosas, pues ya lo volví a retomar. Y yo pienso que ahora digo, qué bien hice en volver a retomar este trabajo, porque esto pues estaba casi por desaparecer. Y sobre todo iba a desaparecer porque, perdón, porque empezaron a trabajar con 2, 3 personas que quedaban con los materiales que, pues por ejemplo, el yeso, que no sirve para este trabajo. Y nosotros pues ahora estamos dando cursos. Estamos dando cursos porque yo quiero dejar esta pues herencia, ¿no? De que a lo mejor de 25 que tenemos o 20 que tenemos de alumnos, pues a lo mejor 1 o 2 personas van a decir que les va a gustar, pero que lo que yo no puedo pensar que pues no va a pasar nada si desaparece, ¿verdad? Pero qué malo. Sería muy malo que se terminara esta artesanía que es muy emblemático para Michoacán, para Uruapan, es muy emblemático, porque es una artesanía prehispánica y que pues es la herencia de los purépechas. Y como yo soy purépecha, yo soy indígena, soy purépecha, hablo purépecha, igual que mis hijos, hablamos purépecha. Y bueno, pues ahora estoy muy sorprendida de escuchar tanta química.
Yo pienso que también es muy importante esos encuentros porque nos permite mejorar nuestra técnica. Y yo, por ejemplo, hoy aprendí muchas cosas de por qué unos utilizan aceite de linaza, por qué otros no, por qué otros utilizan aceite de chía, y eso depende de las tierras y de todo eso. y es importante también que nosotros hagamos un buen trabajo para que ustedes no tengan tanto trabajo para la restauración.
Thank you very much. It's 5 o'clock and I think we're about to turn into pumpkins, but in closing, I first want to thank everyone for their attendance and their presence and participation. Above all, I want to once again acknowledge the decisive support provided by the National Endowment for the Humanities, which made it possible for an international, interdisciplinary group of researchers, scientists, conservators, and art historians to fruitfully collaborate on a project that advances our capacity to identify, analyze, and characterize the physical and mechanical properties of chia oil as an artistic material. This groundbreaking work not only has implications for conservation and preservation practices, it opens new avenues of research that productively challenge and expand the disciplinary boundaries of both technical and historical analysis of artistic material engagement. Paradoxically, in the laboratory, materials are isolated. They're literally extracted from historical contexts of making and meaning. In the case of chia oil, a binding medium that is only visible as the carrier of color and form, scientific analysis can make its properties knowable and interpretable. Traditionally, the artist's workshop was and is the site of a vernacular understanding of the properties of chia oil. It's a place where its usage relies on skills and technical knowledge transmitted, often intergenerationally, through training and practice. In other words, workshops like the one headed by Juan Correa in Mexico City at the end of the 17th century were not just sites of labor and production, but locations of knowledge, where artists made informed decisions about what materials to use based on their suitability, availability, and cost, among other things. Insights into these artistic decisions and technical processes are sometimes, albeit rarely, revealed in written documentation, some of which was produced by the systematic information gathering of chroniclers and colonial officials for the economic benefit of the Spanish state, and some of which simply documents the mundane consumption of materials. Think of account books, shopping lists, etc. The findings presented today call for a close and more critical and nuanced reading of written sources, and above all, for the renewed study of things. The art objects that are preserved in museums and cultural heritage sites, using a toolbox that now includes novel methods of analysis, such as proteomics and genomics. I think it's fair to characterize the research presented today as work in progress, not because it's unfinished, but because it encourages new insights and points the way to possible solutions to long-standing questions. Thank you very much.